And so I was here working with the first sequencer, the Select Illumina sequencer on campus that was in the Nelson lab, the GA1. From then, every run that we do now on the Nova, is like 1,000 of those runs there. More, every run we do on the OSEQ is more than the entire history that that first machine produced. In the beginning, we'd like to short number of reads you get per lane is so small. Of course, you're not going to run one sample in a lane. In fact, our first big project where we with the Tech Lab, we developed the BSC, and so we did the first single base resolution in methylome interrapidopsis. It took us like 64 lanes or something on that GA1 to cover that in a single background wild type. Wild type was like 64 lanes. And we had to run like 120 lanes to get 60 lanes of usable data because a lot of lanes had trouble. Nowadays, that experiment is just a tiny, tiny fraction of an S4 lane on an OSEQ. And so, of course, you now have to make, we've reached the point where for many experiments, the amount of data you get from one lane, instrument is way overkill. And so rather than waste money, you'd rather run multiple samples. And so that leads to multiple. And multiplexing is now completely routine. It's essential for efficient use of the instrument. Most people think of it, I think of something so simple that it's not even worth discussing. Oh, it's so simple, it basically never really fails, and it's spectacular. The data we get out seems okay. It must be working fine, nothing to worry about. But actually, that's not really true. And I don't know if I'm going to be able to show evidence for all true, but at least I'm going to be able to tell you from personal experience. I did basically Dmacc for every OSEQ plane that the BSC or C16 core did from the beginning of the week till about the middle of last year when it just became completely unsustainable for me to do it. And so I did about 600 some lanes. And I did those lanes without the typical assumptions people make, which is basically that the sample sheet that you come up with ahead of time is correct. And the process basically works. If you drop those assumptions and actually look at the data and work from the data up, let the lane tell you what's there and then try to match that against that equity, you find that there's a lot of things that happen that don't really meet that model. And so, like I said, I'm not going to be able to maybe show real hard evidence for basically everything I'll talk about has been pretty usually at a good rate, a few percent, 5–10% sometimes. And the impacts range from moderate loss of representation to pretty severe. Some that'll be kind of obvious, or at least you'll have hints of it with the usual approach, and sometimes the usual approach will just be completely hiding it under the surface, and you won't even know unless you kind of go looking. So that's what this workshop is about. It's sort of having to go find them and then having to remediate them once they happen. Well, if you can just avoid them from happening in the first place, like so that's really the main approach that kind of I want to communicate is simply by the end of day 2, you'll have some short list. Don't do things, and you're probably in good shape. If you are dealing with already existing data, or you can't really do all those things because constraints of your experiment or whatnot, and okay, then maybe you got to maybe at least have some awareness of what problems can occur, and so on, and then kind of in ascending levels of complication, then actually able to find in the real data, and then even more complicated, being able to remediate it in real data, and so on. So, I mean, there's different levels that you can enter, but even if you just kind of take away from this, just the kind of things to avoid, that'll be good, I think. So, yeah, so this is sort of the rough plan here. Today we have to build up quite a bit because you have to kind of to really appreciate what's going on and have some insight into why maybe some of these things happen. You have to really kind of understand these features pretty well. So we have to talk about the Illumina platform and kind of how it works inside, and so there's some details to go into there. Then we'll talk about some of these Dmacc myths that people have. Sometimes even kind of really not true. I'll kind of talk about this blind define what people do, which is kind of blind faith in the process of sample sheet versus the sort of bottom up de novo approach. And then we'll start a worked example. This first worked example doesn't really have anything fabulously wrong going on it, but just to kind of start kind of with a simple case before we can talk about more complicated cases. And so that'll bring us to the whole process of what raw data really looks like off the instrument, how that data gets converted to FASTQ files that downstream uses. We've got a lot to talk about in there. That'll be most of today in terms of time, I think. And then day two is kind of we'll talk about dual indexing. Today we'll probably mostly single indexing. Dual indexing, it brings up the idea of index hopping, which surely happens in single links. It's just you can't really observe it correctly, whereas it's dual link, it's very obvious that it's happening. UMIs, what do you do when you try to mix libraries of different index types in the same lane? A little bit about phi-x, which is something we used to spike in routinely now because of reasons. There were also reasons to include it in the first place, but after experience now, and then the main thing on day two is this problem zoology. We're sort of about the universe of things that has actually been observed. And then the big summary there, what not to do, right? Things that sort of push on the soft spots of the technology and aren't, that kind of are, if you can avoid those, you're probably going to be fine in Dmacc, even if you don't really look hard at it. And then custom index design, because basically some of these problems are due to the fact that commercial kits, even commercial kits from single manufacturer, say Illumina, they're not well designed currently in terms of indices. I don't know why. I think there was no coordination, and some of these kits come from companies that Illumina bought and whatever, and once the indices are evaluated, and they weren't necessarily combinatorially optimized in the beginning. So anyway, if you do have the opportunity to do custom indices, then you can actually do better than some of the current designs that are just super common just because they're in kits. So I think for the first about 2 hours, so the schedule, I think, blocked out 3 days, 3 hours. That's a long format February workshop. I don't think I really want to take that much time because, I don't kind of know people's interest and what level of detail you guys are in to this kind of stuff. So I think for this, this first time, the plan is sort of came more closer to 2 days, 2 hours. And I'll kind of reserve that third hour for either spill from kind of the plan for the day. So I'm not really sure whether today's going to kind of take closer to 2 or closer to 3. As I said, this is the first time I'm doing this. So, it's always hard to kind of figure out how long things take without doing it once first. And then sometimes we get questions. If the questions come over Zoom and some of those are kind of hard to deal with on the fly. So anyway, longer questions or Zoom questions we'll handle with that third hour kind of thing. But if you have shorter, of course you have questions. There's not many of us here, so if you have questions, just just ask. After all, the only reason to do this is to help you guys, right? I already know this stuff. This is all stuff that's deep in my personal experience. So I don't need to. Myself, I already know all this stuff. I'm trying to communicate it. So, the point is to help you guys. All right. So maybe for a bit here, we can run with the full screen slides. Maybe make this chat window smaller. Okay. Oops. No, there we go. All right. So there's a million zillion applications of sequencing now. So we have to take, in terms of getting anything done at this work, we have to take a very cartoonish view of individual applications. So, we won't really want to get into, if we got into like Chromium 10X today, thousands of all the kind of complicated multiplexing situations and so on, we never get to what we kind of there. And basically for what we need to know in terms of this overall idea of Dmacc and how it works and how to approach it and problems. So we don't really need to know the details of the application so much. So our view of the sequencer is kind of like this, basically. By whatever process, you get DNA in a tube, right? Lots of molecules in there. And the sequencer's job is to basically make some random sample that uses billions of the sample size is billions. In the old days, it was hundreds of thousands or millions. Old days for me is like J1. Old, that's my generation. Older people, of course, would go to Sanger sequencing, or the days you would run gels, for like 100 bases or something long or whatever. Anyway, but from my point of view, the old days is like J1. So because this is what a NovaSeq looks like. This is a NovaSeq 6000. It's sitting in Karasaki, the one for the PSRC in the Karasaki third floor near the elevators. The goal is to just, this is relatively short pieces inside this too. This system cannot handle long pieces. So, the reads are going to be fairly short. Like I say, in the GA1 days, that was 27 bases, 36 base reads. These days, you could have Paradigm 150, 150, or 250, and so on, things like that. Some of the other platforms these days, Nanopore and so on like this, they can do much longer reads. In fact, maybe you saw the science paper recently about the proof human genome that was done using long read platforms, filling in most of the gaps from Sanger. We're down to like 2 gaps left now. So the reference genome. But anyway, those platforms can have very, very different error profiles, much higher error, and much more complicated. Anyway, but this is basically our picture of the Illumina sequencing. The evolution of the platform over the last 15 years or whatever has mostly just been how many of these sequences can you make and how long are they, right? But basically, that's the idea of this thing. So here's the major generations of instruments that we've kind of run. GA1 around 2007, GA2, 2X, Hi-C4000, 2500, Hi-C4000, and now the current NovaSeq 6000 we run. That one around 2017, that's been a while. It's probably near the time when we moved to another new instrument. If you've done any sequencing with our core and even the JCCC core, NeuroCore, or whatever, I think we infected them with our naming system. Our run names, lane names tend to be like this, like ZDP 160L2 if you've done any sequencing. That Z is the instrument name. The instrument, we use a single letter for every instrument. The D, in this case, tells you which kind of flow cell, which throughput capacity flow cell we use. The D indicates an S1 flow cell. The machine, also a NovaSeq 6000, like several of the previous instruments, has an A side and a B side. We can physically load 2 flow cells on it at a time. And the D tells you it's on the right side, the B side. The 160 is the 160th run in the Z. This is the Z S1 series. The P tells you it's a paired end run. And the L is for lane, and that says lane 2. These S4 flow cells have 4 lanes that you can theoretically load independent library mixers on those 4 lanes. And I think I brought an S4 flow cell I can pass around. If you want to see what one looks like. There it is. Oh, and I think I threw in a Hi-C4000 flow cell here. The Hi-C4000 is in this tube, the big guy, which makes sense because one way to make throughput higher is simply make the surface bigger, right? So you just have more area. So that's the S4 flow cell theory. If you get the light to bounce off it, you'll see the refraction pattern because this thing is nanostructured, nanometer scale objects on this thing. Visible light, being around 500 nanometers, whatever, diffracts, and so you'll see the rainbow goes off this thing. You can take it out of the bag if you want to wash your hands. There's chemical residues on these things and so on. But you should wash your hands anyway all the time with COVID and stuff. But yeah, anyway. One of those, a NovaSeq run is, an S4 run is like $10,000. So course, once they're done, it's garbage. But but anyway. All the flow cells in the platform are exposed, and basically all sequences exposed. Well, it depends on what you call a unit, right? You have to buy not only flow cells, but the actual liquid reagents and so on that actually get cycled out of it and so on. But I think order $10,000 for flow cells and so on. Cores, we're not making much money. We're generally losing money. Most of the sequencing cores are, in one way or another, somewhat subsidized. If usually, so if you look at the cost you pay for a lane, multiplied by the number of lanes on the flow cell, the costs are usually even. Maybe that covers reagent costs. Sometimes, it may not even. Certainly doesn't cover personnel costs or instrument depreciation. These are $500,000 or more. Service contracts alone can be $50,000, $100,000 a year, plus the reagents and so on. I mean, these things basically just burn money, right? They just burn money. But yeah, I mean, we're not, as expensive as you might think lanes are, you're actually getting fantastic deals relative to what the cost would be if you had to buy an instrument and people to run it and keep it going and so on. So anyway, so so you see the evolution kind of the throughput, low millions, now we're into billions of reads per flow cell. Lengths of a read used to be 36 bases, or even started lower than that with the GA1 first version. And now we're into the 150, 250. I mean, everybody would like that to be much bigger, kilobase, whatever. But the Illumina platform, Illumina's got this thing that they're going to say, oh, we got long reads coming this year. Well, we'll see what that means. Paired ends didn't used to be a thing. That was one of the things GA2 added. The number of lanes for flow cell has always been, like 2, 4, 8, something like that. Usually runs are a day to a week, something like that. And so, but here's the main point of why Dmacc became a thing. It's because somewhere around the Hi-C or so on, things got big enough that it started to make sense to put multiple samples. Well, so that's the easy way to make higher throughput. Simply make the surface that's participating. You're not going to make any technological improvements. You can simply do what you're doing more of it. Right? And to some extent, that is why that's where that little flow cell in the purple tube is the Hi-C4000. That is a fairly similar technology to the Dmacc. The Dmacc is dialed in a little more perfect. But basically, those two technologies have basically already reached the point where since it's a fluorescent-based system where you have fluorophores on the HCGPs, and you're using visible light, from microscopy, you can only image so small. Visible light has eventually, you get down to the nanometer scale. That's it. You're not going to be able to distinguish separate spots of different color and so forth with light at that frequency. And they're almost there. But with the Dmacc spacing of the wells inside of the well, the optic system is already basically at physics, the limits of physics due to the wavelength used. So that's basically one reason why the export flow cells had to go bigger, because it's bigger surface. Because you've already kind of operating with a given kind of technology scaled down to the limits of physics, you're kind of that's really quick. The earlier technologies weren't as optimized as this. The earlier systems didn't even use what are now called pattern flow cells, random growth process. That's not quite as efficient use of space and so on. But yeah, that is indeed one of the ways the Dmacc got higher throughput, simply the use of bigger surface. Okay. So the Dmacc6000 has 4 flow cells, right, based on capacity of what you want to run. Those are known as the S prime or SP, S1, S2, and S4. Roughly in terms of throughput, that's a 1x, 2x, 4.5, and 6. We will talk in a minute about how these are physically divided, because that shows up in the FASTQ file, the actual physical position. Every FASTQ read tells you essentially the physical position on the flow cell where it's read. Most of the time, you're not going to carry it downstream, but for some of the stuff we're going to talk about, we actually sort of do want to know. And so there's physical geometry for each of these. In fact, most of these are, in fact, are imaged on both surfaces. What's a flow cell? A lane of a flow cell is essentially just a glass box or a silicon box or whatever, but it's essentially just a box with two holes in it. And in one hole, liquid can go in, and the other hole, liquid goes out. And it's called a flow cell because then the liquid flows in the inside in a narrow channel usually. And actually, usually, good thing they did the system this way. There's two ways to do it. You can either shove fluid in here under pressure and it squirts out the other side, or you can negative pressure this side and suck it through on this side. Much better system to have it sucked through because pressurized systems always leak and whatever. I mean, so much better. You lose seals here or whatever. Simply liquid doesn't flow. You just suck air on the sucking end. Whereas if you're pushing, you got a method. But that's all a flow cell is. And in fact, they usually image the top and the bottom surface. The top is transparent, and if you can just set the focus of the microscope objective basically. Either put the top surface out of focus and aim at the bottom, or just aim directly at the top surface. So you usually image the top and bottom. It's in long strips. In terms of the camera, of course only sees a tiny space because you're not going to make a camera with billions of pixels. Not at prices anybody in normal human for it, or where normal human right now is like a major research university. So of course we do that by using a high quality camera sensor, which we then drag across the flow cell to get huge numbers of pixels out of it. We don't really need high frames per second here, right? A frame is like 15 minutes or something like that. That's what a chemistry, imaging cycle is like, something like that. So we don't need high frames per second, so we can essentially make our camera super high resolution by just dragging the sensor around. And so we basically just, the image sensor is only covering a tiny little piece of this, and so we divide this area into strips and little segments and so on and number them all. And so we essentially divide this whole thing into little squares that the camera answer, with one picture for each square. And so this geometry of swaths, which are the strips, and tiles per swaths, which are the divisions this way, is the tile division. And then each of those tiles has millions of wells in it, which are then numbered in a particular way. And all that becomes the read numbering that comes out at the end. All right, so how does the actual sequencing work? So you may know that Illumina's platform is this SDS, sequencing by synthesis. Essentially, we co-opt nature's DNA replication. We use a polymerase, free nucleotides. The free nucleotides have fluorescent labels on them. And then they also have reversible terminators. The idea basically is that you're going to do millions of reads in parallel, but they will all be at the same relative position at the same time. First base for everybody, then second base for everybody, whatever. So it occurs in cycles. You get everybody on the first base, blowing their color, take pictures, kill the terminators, incorporate the next base, which is only one base with the terminator. Then image everybody for cycle 2, and flow that away, kill the terminator, cut the fluorophore, do one more base, and so on. It just works in cycles like this. So that's why your prepared target's got to get these adapters on them, because these adapters have the interface to the flow cell. The flow cell is coated on all sides with oligos of some kind that provide the binding sites where the dye goes. Here I'm showing the old style random growth process, but the nanowells are almost the same. It's just that we control better where things happen. But the idea is basically, these individual molecules are tough to observe, right? Single fluorescence of a single molecule, very, very low intensity, hard to observe. So the idea is always to turn one of your dyes into a macroscopic observable colony, maybe 1,000 copies or something like that. So each of your library dyes lands somewhere on the flow cell, grows up into 1,000 colony, which is hopefully all copies of the same thing, and then hopefully the chemistry keeps that colony always in perfect sync. But basically that turns your little almost infinitesimal unobservable signal into a macroscopically glowing spot that we can handle with technology. Okay, so there's basically a polymerase reaction, some kind of amplification reaction here. In the old days, it was called bridge amplification because of the little bridges that they have to make to get the two end poles attached to the flow cell and so on, keeping things tethered physically. You can't really do this amplification in liquid phase because that would end up with everybody everywhere. You have to do amplification in a way that things don't spread out physically, that they all happen in one place, so that you get your spots that are observable. And spots correspond to individual molecules of your library. So anyway, now it's XAMP with nano wells, which we'll see a slide for in a second. But basically, we grew up our little colonies. And then when sequencing starts, you've got a primer of some kind that gets, controls the place where you start sequencing. The primer will align to, one end and the other will be doctors right at the boundary between the doctor and your sequence. And so cycle 1, when we flow in the first plus they label 3 nucleotides, will just go flowing at the first base of your insert. And then we image the terminators on the fluorophores, make sure that the polymerase doesn't proceed. We capture the first base from the whole flow cell. Now chemistry kills the terminators, kills the cuts off the fluorophores, leaves the nucleotide there so that the position 1 is used up now, and effectively the primer is now 1 base longer, right? And now base 2 comes in, right? And now we expand. Right? And you can imagine so many chemistry cycles like this, like whatever, 10 reagents you've got to flow for each cycle. Maybe they're 99 point, these days 99.99% effective or something, whatever. But you're going to compound those chemistry cycles over and over and over again. By the time you're at the 150th base of the of an end, maybe things aren't so good anymore, right? You've got the colonies not all in sync. Some guys are on base 149, some guys are on base 151, and all that becomes important for the base calling and so on. And it's the reason why it's not like you get perfect base calls out of this thing. Also, the old chemistry used to be 4 colors. The 4 fluorophores literally had 4 different colors. There were 4 cameras that took the 4 wavelength filters, 4 color filters, 4 grayscale images. The new style chemistry is called 2 color. We just use red and green basically. The illumination is always by laser. The red laser and the green laser basically, and now the 4 bases ACGT are more or less red, green, yellow, which is both of them, and black, which is nothing. So it used to be red was a color, green was a color, A was a color, C was a color, D was a color, Q was a color. Now it's coming. This actually has observable effects because things that are dark, things that don't prime, and so on like this, actually now reliably call it poly G. So poly G is basically really what it means is dark on the flow cell, which can happen by not just because sequencing is occurring, but that's because no reactions are happening. So you may have, for example, if you had some custom indices or something, your primers aren't right. You get to the index cycles, your guys don't prime. Well, now your indices will actually read as poly G because you're just not flowing during the index. Okay, and things are all more complicated than I'm really talking about here. The colors have interactions. It's not like it's pure red, pure green, totally separated, or whatever. It's just, we're talking in very round numbers here. This is what the pattern flow cells look like. For example, the NovaSeq platform. Instead of randomly having your guys land on an oligo log, the flow cell is actually made with nanolithography processes like we do with computer chips. So we can make billions and billions of these perfectly formed, essentially nanoscale structures. This is cross section. This is the top view. That's the deep view. The wells are coated with the oligos now, and the idea is that when you wash your library over this thing, and your guy will find one of the oligos in a well, and then due to the way the dynamics are set up, the polymerization reaction hopefully stays confined to the well by the physical boundaries of the well and the fact that the top surface of this thing is ground. So there's all these surfaces here are sterile. And then the reactions in here are very fast. And so essentially your guy finds an oligo and then whoop, it just polymerizes instantly and the well is filled up. And then, so if somebody else tries to get there, it's already used up. That way you don't get multiple guys in a well. And it also allows you to kind of watch decently high concentrations up here so that you have a chance of hitting all the wells and filling them up. Because empty wells are useless and waste. So this is the XAMP, exclusion amplification. That's the modern system is based on, and it's quite nice. You don't have all the business of trying to figure out where the clusters grew and clusters overlap and whatever. Everything's nice and separated. And the XAMP gives you theoretically and in practice some concentration invariance. The percent PF1 here up at the top, this is the percent of wells that we typically find singly occupied, so essentially usable wells. 80% is typical, maybe the common range is 63 to 90%. once in a while, we'll have somebody hit 90% utility. Those are, that's like a super run. But you can see there's a left tail there where some runs didn't, some lanes didn't do so well. But if you find yourself in the 80s, you're very typical. 60 to 80, gets the low side typical. You still can't complain too much. Of course, by the time you get to 50% versus 90%, there comes a twofold variance in throughput. But anyway, but that's the typical distribution you see. So PF1, PF0 is, there's a history why it's the PF, it's the purity filter. That was in the days of GA when there was also a chastity filter. For history, it doesn't really matter. But essentially, PF1 is the term that, in terms of pattern flow cells, it meant different things in the earlier days. In pattern flow cells, PF1 means singly felt wells, and PF0s mean empty wells. There can be a few multi-wells. They're very rare. Basically, something you don't have to worry about. PF0 basically is almost all. You don't really have any control. And XAM codes are pretty good job. There's so many other problems that are bigger magnitude that really you don't have to worry about. It's really not. There are other problems you got to worry about. There's a long list of problems before you should fix it. Most lanes or libraries have problems that are worth. Not a big deal. Yeah, so you don't watch. If you watch like 1,000-fold too high concentration flows out, who knows what happens, right? Because then you're so far away from the dynamics of the chemistry in the well are aimed at a certain level of concentration. Right? So if you increase the concentration of your loading orders of magnitude beyond that, well, now maybe you do have multiple guys hitting the well before the first guy could actually finish polymerizing the well. Now you got. But that is so far away from the recommended loading concentration. But maybe when people make errors of loading, it's like tenfold or something. In the core, we load pretty accurately. Whether you quantify your stuff accurately is another issue, and whether everything that quantifies in your library is actually usable is another. So that's still why we get cases where people are on the low end of the spec. But usually overloading is not really a thing. You have to make a real gross mistake somewhere to try to fix that all. And even with all the mistakes that we have, those don't. It's not really a problem class we worry about. Okay, here I'm showing the Hi-C4000 layout. As I say, it's basically the same system with the Nova sequence on. It's just the numbers are a little different, but it's the same idea. So if you say basically flow cell reagents go in one end, sucked out the other end. A lane is divided into imageable swaths, which are then divided into sections that just make them roughly square so that they're easier to process as a 2D image, or parallelized processing or whatever. Logically, it's just all, it's just all sort of one gigapixel image that's just in pieces, right? It's more convenient to process in pieces. But basically, so there's top and bottom. Where this guy, it's left and right. The Nova can have more swaths than that. But the strips are the swaths. These are the tiles. And then the numbering that you see in the reads is related to this tile. I mean, the first digit, like the tile can be 1101, 2228, something like this. That first digit is the surface, top or bottom. The next digit is the swath. And then the last two digits are the tile within the swath. For the NovaSeq, what the tile number you get is going to depend upon the flow cell. Because basically the tile sizes don't change in the NovaSeq. Whatever these little squares is that's considered one imageable unit is always the same with the exact same nanowell layout. The variation between the flow cell types is simply how many of those units there are to go in it. Okay, so Illumina does not actually document really the association between the numbering and where it is. So is it like standing at the front of the instrument, left hand here, whatever, front, Is the 22 is the 1101 top left, top right, bottom right? You know, which one's the top, which one's the bottom? Illumina doesn't really say. I mean, they know for sure. Their engineers know, somebody in Illumina knows, but they're not saying. Yeah, you still got questions of left, right, flip, and so on, whatever. But basically, because there's this thing called XAMPP duplicates, which is another thing we'll talk about on day 2 when we get to like UMIs and duplicates and stuff. XAMPP duplicates, which people mostly completely ignore and isn't really, I used to check for it. I don't really check for it anymore. It never was a huge issue, and I think for many, like, if duplicates are a problem for you, you probably have like PCR duplicates and things that are just so many orders of magnitude higher. You're going to have to do something else. Not, you don't need to directly address. If duplicates are ever worried for you, you've probably got duplicates from other sources at orders of magnitude worse than this, and so you're going to have to do something else to take care of them. You want these to take care of themselves to do that. So it's not. That's why it's not really a big deal. But basically, yes. Sometimes during XAMPP amplification, a polymerizing guide can escape his well and flow along a little more and polymerize another well. And so now you've got correlated wells, essentially different wells that are copies of the same original molecule library. So in fact, if you kind of carefully search for these things, you can find that the duplicates actually exist, of course, higher at very small distances, but extending out into very large distances, appreciable fractions of a flow cell, into sort of a comet-shaped trail where the comet tail is biased in the direction of fluid flow. You can actually observe this. But one positive thing that you do is actually reconstruct the tile numbering. You can actually just see, you know, this is a locality-based effect, so it tells you which tiles are close to each other. And so you can actually rederive, you know, this up to, you know, flips and whatever. But basically, that's the X goes in the swath direction, Y goes in the long direction. In terms of coordinates, and the numbers go like that. At least for the Hi-C4K, it's more complicated. I mean, the 6K, there's more formats to talk about and stuff like this. So anyway, so within each of those tiles, like I say, there's a honeycomb pattern of nanowells that are given XY coordinates. The system depends on whether you're using QC or fast view, nobody uses QC anymore. There's a 10-to-1 ratio in the distance units between them and offset. But basically, for the 6K, you notice the 6K numbering, the X's go from about 1,000 to about 33,000. The Y's go from 1,000 to about 37,000. There's 1776 exactly columns. There are 2304 exactly rows. That makes the wells per tile exactly 4,091, 4,091,904. And you know, there's the square root of 2 and stuff involved in square root of 3s and honeycomb, but everything's equally spaced basically all that, because the integers are only got 2 digits here. You know, there's some nominal uneven spacing. Basically, that's where the XY coordinates come from in your FASTQ names. Okay, let's move on. Okay, so here's our cartoon view of libraries and one. So by whatever method, you know, there's so many applications of sample prep these days. You've prepared in library for each sample. Library is just, you know, hopefully billions and billions and billions of distinct molecules that you started with from your biosample have been gone through your sample prep and now exist as basically adapter, sequence of interest, adapter. The sequence of interest hopefully is, hopefully and typically is maybe a few hundred, low end of a few hundred bases, right? The adapters are dozens of bases. Those adapters have 3 basic jobs. They're your interface to the flow cell and the amplification system, XAMPP. They control what parts of your molecule, either parts of the adapter or parts of your fragment of interest, that sequence at what point during the novoseq run. And they serve as an identifier so that if you are mixing multiple samples within a lane, you know which sample for each thing. That's the 3 jobs that adapters have. Now, how do runs go? Well, these days, there's, you know, you get basically 4 shots at reading, up to 4 shots at reading a particular library molecule. Of course, the main one, you know, and the first one that was ever, you know, in the GA1, that was your only one, is the single and first main read, right? You prime. And in this picture, don't take the picture too literally here. I'm being very sloppy with forwards and reverse complements. I'm being very sloppy with kind of how the priming of the adapters actually works and so on, but kind of the picture, the mental picture is something like this. It's a cartoon just to appreciate kind of the, it's enough to interpret what things happen, but the actual chemical details and so on do not necessarily reflect exactly what the single and primary, you know, right at the boundary between the adapter and the diode, and we build out the first main read, which is maybe 51. These days, typically 5100 one cycle, something like that. The reason why that's not exactly why that's 51 and not 50 or 101, not 100, we can talk about, you know, in the third hour if you want or whatever. It's related to desync corrections and the fact that you want we guarantee 50 bases, so we have to do 51 so the 50th base knows what the 51st base is. Don't worry, it's not a big deal. You get a free base if you want to think of it that way. It's just that the 51st base is a little lower quality. Then you have the option to read some piece of the adapter. Well, I mean, these are priming events are involved, so you know, whatever. If your real insert has, if your real DNA fragment of interest has some magical sequence in it, maybe you get something there too, or whatever. That's another thing to worry about, I suppose. But basically, you hope that in this priming, it's some repeatable piece of a particular place in the adapter, and you read out the index read, so you can embed into the adapter some code that identifies the original sample, and that's how multiplexing and demultiplexing will work. And then these days, you actually get a second shot at reading another into the adapter so that you can do what are called dual indexing. And that primes somewhere else, or I don't want to get into the details of all the variants, but logically, it sort of could prime somewhere else, and it's another piece of the adapter. The details of that have changed over time, including even in Novacy. There's a 1.0 chemistry and 1.5 chemistry. And for example, those two differences change the way Phi-X I2 prime. And that's because Phi-X isn't really an index library. It was never designed with the current adapter indexes in mind, and it just does what it does. Then you get the shot at reading the other end of your insert, the PE main, the main read for the other side of your insert. And of course, there's all kinds of things that happen. If your insert is very short, the main reads can read past into the adapter. If your insert is very long, the main reads won't touch. If your insert is between those two extremes, the two guys can overlap and not reach the adapter, and so on. Okay, but basically there's four ends, right? There's main read, I1, which I'll call, SE for single end, even though it's the first end of a paired end. I just want simple names you can just use over and over in this talk. So SE for the first end, I1, I2 for the two ends, and PE for the second main end. And that is the order that they're done on every Illumina platform to date. Not every Illumina platform did all of those ends, but the ends have always, for the whole history of the platform, always been in that order. That's the order they are physically collected. And that actually ends up reflecting in the class queue file names too. So there's R1, R2 for read 1, read 2, I1, I2, read 2. They're just numbered based on the physical time of occurrence. The first read is literally the first one to physically have been read out. Typical number of cycles we run these days are first main end, and I've put them in the most popular to least popular order here. 101, 150, 151, of course you know those. Then there's weirder ones, 122, 28, 111, 76. These are all basically related to particular library protocols that are a little unusual. First index read typically these days is 8. 10 is also common. The EMI is 16. 7, 9, 17. The 7 is really 6 plus 1, because the first indices, which are still around the true sequence 12, are nominally 6 words, although I don't recommend using them that way. We should at least consider them 7 words. We'll talk about that when we get to the empirical Dmacc. The second index read, none is a very popular choice because people feel was single indexing. When it is, their most popular is 8 and otherwise 10, but 24 would be involving UMI probably. And then the second main read, pretty much the same thing as the first main read. But some of these other ones, like 91, are paired with some of the other weird ones up in the first main end for particular protocols. So if you actually put them together and ask for the whole geometry, the most popular one we've been running recently is 101, 88, 101. The next one would be 51, 88, 51. 118, 101, 51, 51, 10, 10, 151. And so on. Either some more, like 51, 8, 24,
You know, 101, 17A, 101. We have to find one of these 20 guys or whatever. 28, 8, 91. I suppose I should have said 28, 8, 0, 91. No, I guess I didn't do that. And then there's a question of how many samples are people intending to mix within a lane? As I say, they're not always correct because empirically you can, when I do my empirical resolution, you find out that there's often differences. I don't want to say often, it's not like every lane or majority of them, you know, get for the purposes of this talk, 5% occurrence is often. This is the number of samples that users are intending to mix. You know, single-ended indices 3, 152, it's the range. This is the high end currently. You know, 192 is the most we've seen lately. I think when we started NovaSeq, we had like a 300, 400 plus or something. It just depends on what you're doing. But this is sort of a common range. This is, I think most of this I'm going to show you is in the last year is the data frame that I collected for the workshop. But this just gives you some idea, you know, where's the median, right? You can just kind of see that by eye, right? Well, the median for singles is around 20, and the median for duals is point your finger in the middle, it's in the 30s, maybe a little higher. And then unique duals also make it 30. So, you know, 20, 30 maybe is mainstream, not of multiplexing right now. All right. And we'll need to know a little bit about the image analysis the base caller needs to do. You now sort of understand the raw data that the instrument is actually collecting, which is essentially these grayscale intensity images for red and green for every combination of lane, surface swap, tile, XY coordinate, and cycle. So there's some gigantic matrix, or at least the mental picture of some gigantic matrix is being collected. This is terabytes of data, typical S4 run. You know, where it's basically we just get these two intensity numbers for this huge tensor of indices. So the job of the RTA base caller, which RTA is a piece of software that lives on a Linux machine that's embedded into the instrument, and essentially the raw data, this level of data, the actual images and the actual intensity matrix like this never leave the instrument anymore. It used to be. In the GA1, in fact, the instrument didn't do anything except collect images. We literally got like TIFF images, like a gigantic record of TIFF images. And then you ran, they gave you some source code, you compiled that, and then you actually ran the pipeline yourself on the block image. And eventually they moved on to real-time analysis. The RTA means real-time analysis. So it's much more innovative than the instrument. You needed a pretty beefy machine to do it. And this Linux machine inside the instrument works hard. It's a pretty beefy machine. This is a big job. But the RTA base caller basically take this thing and come up with the ACGT and ends. Right? It rarely takes calling ends, but that's the that's the decision. Is basically turn these two numbers into ACGT and then you've got the per base Fred scale quality score, which is its confidence that base caller is correct. And you think, well, what's there to do? You make some scatter plot of these two things, call it the find the 4 clusters, draw the boundaries, done, right? That wouldn't be completely wrong, but it wouldn't perform very well on it. It's a much more complicated than that because the chemistry is not perfect, molecules are desynchronizing over time. So to interpret this number, you sort of need to know what the interpretation of the numbers nearby is. The physics of visible light and nanometer scale, then it's not like the optics are perfect here either. You can get bleed from nearby stuff and so on. And, it's not like we want to pound these things with the lasers either, because if you pound them, it's the same thing you get with, whatever, a confocal. Eventually you bleed things. I mean, you put enough laser power into something, and it's not happy. Right? So, and then the laser power is in fact ramped up towards the end of the run because as things kind of decay. And sort of parts of the colony go away, don't work anymore, and the chemistry fails. We actually do hit it with intense laser light, more intense laser light at the end of a run to try to compensate for the thing. Runs there's just chemistry. You're doing chemistry, right? There's temperature variation. It's all kinds of stuff. This is a chemical, mechanical, physical system. There's all kinds of things. And so this is very complicated inside. There's a lot of hidden, we haven't seen the source code for RT in a while. If you've been around since the GA1 days, you still have a pretty good idea of what's going on down there. That's the problem. But the details of exactly how to do things is going not public. And you can imagine there's all kinds of things in there where there's auto calibration based on data and so on. And so one thing that maybe you can already appreciate is that if, for example, especially in the first few cycles of a read, your base calls, your data is all very skewed, then things that auto calibrate based on the assumption of having a little bit, at least appreciable amounts of ACGs and T. Because to learn ACG and T from the data itself, if you're trying to kind of auto calibrate, the data hopefully provides you with examples of all of those four, right? Because otherwise, how do you know? I mean, you're trying to learn the patterns, and, you're assuming there's four choices to make ACGT, but what if the data is not giving you really all the choices? Well, then you may start, maybe the data only has T. So you're looking at this, maybe the data only has G, right? Which is like dark, right? So you're looking at this and it's like, well, all I see is dark noise. It's like dark, right? So but if the dark has noise, right? It's like you're seeing cameras and background noise, whatever. Well, maybe that's a low intensity thing. So now I got to start subdividing the little cloud around black and say, oh, okay, this is black, but it's in the red direction. This is black in the green direction. Maybe you subdivide that cloud into little pieces and you call, okay, this is the A, B, C, D, B, C, D. Except it's all G's, right? But now your base caller is completely, just kind of making, it's just operating really in the wrong space. So that can happen. So I mean, and the thing is that your experience with main reads being pretty good on the Illumina platform does not translate to index reads being pretty good. Index reads are typically way lower complexity, way more synchronized spot to spot over the same cycle because the number of distinct indices is quite low. Furthermore, that gets compounded by the fact that people mix samples of all kinds of representation difference. It is very common for us to see top sample to bottom sample of the lane 100-fold different representation. And that's not even the end of the distribution. That's just like, not every lane, but that happens a lot. Then there's the guys with 1,000-fold stretch, 10,000-fold stretch. By the time you get to the 10,000-fold stretch between a bright guy and a low guy, well, this guy is shining like a blue giant, like the sun or something. This guy is shining just like a brown dwarf or something, or a white dwarf star or something. Right? The glare from this thing is just incredible. And this guy contributes so little that he basically drops out of all statistical analysis because of auto calibration and so on, whatever. So yeah, index cycles do not have to, your experience with main reads does not translate. Quality main reads does not translate to quality. Index cycles push the platform in its soft spots. And sometimes they go in weird things happen, counterintuitive things happen. So anyway, so the details already have changed many times since 2006. But kind of the idea that problems can occur, that's always been there. But index reads kind of really push the platform and base caller in places that aren't its strength. So we were just talking about this, the fact that if you have essentially low diversity in a cycle, if many wells are all going low diversity at the same time, or many wells are going all the same thing at the same time, that's not good. That violates kind of some of the auto learning statistics of random bases, randomly distributed. And it's not diversity of your indices that matter. When you label your samples, you say sample 1 to n, and these are my n indices, and my n indices are very diverse from each other. That is not what matters. It matters that times their representation. And representation differences can be orders in that. And so if you've got 100 samples, and the sequences are all nicely spread all over verse day, but sample 1 ends up being 99% of the lane, and the rest of the samples compete for the last 1%. That's not a diverse set of indices because the sequencer is basically a lane that's purely the one guy. Auto calibration will go nuts, and all the index base calls for all your 1% guys will be horrible. At that point, it's not like so basically what that means is that guy, bright guy, will now damage the base calls of everybody. So that means that also tells you all kinds of things. That means that basically the Illumina platform is not a linear platform. There are nonlinear effects, and adding things to a lane you damage other things on it. Right? So it is not true that you can take a lane, add a good lane and a good lane, and mix them together and get good. No, no. You get bad. Even if the indices are apart from each other. Obviously, if there's index conflict, that's exactly like I expected this. I not separate no samples out. But even in the absence of stuff like that, if I take a good and the good and put together, maybe not good. In fact, you can also, so that also tells you things like spiking is not necessarily good. So what you want to do, oh, I got this little library, I just want to take a peek at or whatever. Crappy little library, I'll probably do it. Don't do it. We'll talk about reasons why, but no, it's you essentially, the thing you don't want to do in the platform is anything that kind of makes one sample more unusual than others. Essentially want all your samples to be sort of comparable in base composition, comparable in representation, and so on. Whatever. The more you deviate from sort of uniformity, the more likely you are to kind of run into edge. You're going to reach some tipping point. Because of the nonlinearity in the base color, things are usually okay until they aren't, right? And it's sort of hard to kind of figure out where that boundary is sometimes. But it's not like everything just kind of linearly responds to grades gracefully. No, there's usually tipping points where sort of like things are kind of okay. Maybe there's a little hidden problem, then bang, but then things are kind of really, okay. Yeah, I think we're going to need probably closer to 3 hours for the class today than 2. Okay, so now you're kind of primed to at least appreciate kind of things that Michael runs. But anyway, so yeah, what is multiplexing? Well, now that reads per lane are 1,000 times what it used to be, and some applications are well into the point of dimension returns. So we'd rather spend our money on getting more samples than just overkill 50 samples. We need to put multiple samples in all that. And essentially, because we have this index read support now, the machines give us lots of possibilities for how we use that. We can embed into the adapters little tags, sequences. Just tags are just the tamers, 6 or 8 tamers or something. That the library prep or adapter designer gets to choose. For many of us, it's just we just get the text or whatever. But basically, they're little pieces of the adapters that we can preferentially, we can basically attach uniquely to samples. Because when you give the library prep, you can do samples separate, and then you arrange so that each sample gets exposed to the adapters only with the one tag, and then you mix them once all those reactions are done. And you hope that, for example, no active reagents are left by the time you mix samples, right? Because you don't want samples to get tagged with the wrong guy and so on. There's this whole phenomenon of index hopping, which can happen at various places in the pipeline, including on these. But basically, that means occasions where, for one reason or another, by the time you get an index read that looks like, in the absence of all the other problems we're talking about, are more like base call problems. Even if the index read, the base caller is doing fine, whatever, and you get the looks of it perfectly, no base call failures on the whole lane. You get a base call read that looks like a perfect match to a sample. That does not mean it's in that sample. Right? You hope it does. And most of the time, you don't really have a way to tell if it is. But sometimes you can't tell, and sometimes you can actually verify it. Almost all, I don't want to make it sound like, you can't just be multiplexing at all. But no, I mean, it can happen. Usually the rate that happens is quite low. There are cases where the rate becomes noticeable. If you're looking for it, it can happen at levels that are not negligible if you don't notice it, and then it's just affecting your downstream. But basically, it can reach a point where the number of even the index reads that look good don't necessarily mean that's the sample. And if you want to wonder how we can ever verify this, how do you know that ever really happens? Well, for example, you may have a library where you can actually effectively be multiplexed without the index. You may have, for example, an amplifier. For sample 1, actually amplifies only this part of the this sample amplifies this part, this and this one. And that means that essentially when I align the reads in my reference schema, I can know what sample is from because it lines, if it shows up in this region, it's for example, it's free, but this one I don't really need the index to tell me everything. But essentially the genome is my index, right? Whatever. But for those libraries, there usually are indexes. And you can now start to look at how many from this Dmacc file, how many of those reads map to the right region? How many of those reads map to just regions that I didn't really amplify at all? That could be some sort of non-specific thing. But maybe all the samples map to regions you intended, but not always assigned with the right tag on it. That's the case, for example, we have very hard data this and index. Like I said, I can't really give you hard, but I don't have time to kind of give you all the real evidence for how these kind of things. Just believe me when I say that the things I'm talking about aren't theoretical, they happen. The worry then is how to avoid them happening, what rates do they happen at, how bad down and so on. It's not, existence is not a question. Basically, that's the first thing to appreciate. These things happen. It then becomes a question of rates. Rates, how do you deal with it? So yeah, so the logic is basically, unique tag on each of these files, throw them off in the sequencer where the sequencer, these just are floating liquid now, there's no separation. But we get the index reads, and if the index read is a good match here, perfect match, say, and don't know, first 0th order assumption is that came from that fact, right? So we can put it in the file for the right mouse. Maybe one of them gets a single base call wrong, so there's a mismatch here, but it's one mismatch to this guy and more away from this guy, and almost surely this has got to go into mouse 3. Then there's this guy, which is far away from everybody. Well, whatever. I mean, whatever. If it's low frequency, who cares, right? Well, whatever. Unless it's an index that's on the guy next to you on the bench, and it's because there's some cross contamination or something, right? And then it could happen. So maybe not, not everything that's like this is completely harmless or ignorable or whatever, or not. But yeah, but at the moment in this simple picture, yeah, that guy is just whatever. He just doesn't go. Dmacc fail. Okay. Yeah. So we've already talked about some of the, I think most people underappreciate some of the complications can occur here. But the index reads, yeah, they're not. Don't extrapolate from the main reads to quality. People make mistakes, probably more often than people realize, because unless you kind of dig into that file of Dmacc fail, you may not know what's floating around down there. Commercial prep rets, not all terribly well designed from the index point of view. Bias we're talking on day 2, index hopping. We haven't talked about mechanism or really much about it yet, and doing the CPE tools and so on yet. That'll be day 2. And extremely high representation ratios. If I had to say one thing to avoid, it's those. Those are the real killers. Sometimes you can survive a couple of the other problems, but usually, if any problem, that's usually bad by itself. Because, for example, Illumina's default base called error rate is something on the order of 1%. Right? It sort of concentrates those long reads, and you make the indices aren't long reads, so they shouldn't have any problems at all. But no, like I said, indices are different. But anyway. Let's say, without knowing anything else yet, 1% on average. Well, if I put in 100-fold representation ratio difference, that essentially already overwhelms the standard distances of errors, right? So if I had 2 indices that were, say, 1 mismatch apart, and 100-fold representation difference among them, not good, right? Because the one error base cloud of big guy can send 1% of his guys toward me. So that's not good. And I'll make the ratio 1,000. Then you think, well, maybe the error will be distributed over the region, and that won't land on that. They won't all land on base 3 ADT, which is what it takes to make that guy do this guy. Well, you'll see that usually, actually, in index reads, the errors are all concentrated in particular patterns. So they're not uniformly distributed. So yeah, even if there's 1% baseball error, we could be actually sending, maybe not, but if this happens, it can line up, we could send a big screen down towards the other guy's index. Now that other, so let's. Well, I think I have an example. Well, we're about to show a little thought example about that in a second. So before we talk, maybe numbers, just to kind of get a mental picture. Yeah, could that really be serious? No, I think that's in the slide. Anyway, so basically, this is the setting that we think about Dmacc. Dmacc is really an analytical interpretation of the sequencing experiment. Okay. Think of it as basically alignment of very short reads to a very small genome. Okay? The very short reads are the index reads. The genome are basically the indices you're expecting, or maybe if you're a little more general, the indices, all the indices in the kits and boxes you use. So being a little more generous, all the kits and boxes that are used in the lab, maybe at all on the Illumina platform or something like that. But basically, then it's not really surprising then that the Dmacc problem is actually a little more complicated than people are normally thinking, in that you'll have reads that don't align with high confidence. You'll have reads that align ambiguously. There'll be questions of the accuracy of the assumption of the reference. There's a question of how much swap mismatches and whatever to allow in the alignment. And then there's the question of whether the wet bench and XAMPP have really done the right thing by attaching the right tag to the right guy. So the main goals of Dmacc are, in the face of that, to get high sensitivity and high selectivity. We want high sensitivity, meaning for each true molecule from an original sample, it has a high probability of landing in the Dmacc file for that sample. We want to recover as much representation as we can. That's the point of secret. But we also want high selectivity. We don't want to make any mistakes. And in fact, for most people, I think they would say, I would much rather have high selectivity than high sensitivity. Sample mixing is a far worse problem than a little bit less representation. You know, it's not like we have, you know. So I think for most people, they would say, all right, if these ever really, like, give me a little less, but give me a better chance that the samples are selling. Because the alternative, this Dmacc, when things are in the wrong pile, it's like somebody opening all your tubes and taking a pipette. Yeah. So I don't think you tolerate that, right? You don't want the integrity of a sample and it be unmixed with other samples. Cross-sample contamination, that is a big thing you do not want. Whether your downstream is super sensitive to that or not, you know, matters. Like some people have been rare variant detection, like, you know, maybe percent of, you know, at a certain SNP or something, you're trying to find the frequency of copies that have a variant. You know, you want to find rare cases. Well, then maybe even, you know, a little bit of this Dmacc is a real serious problem. It's looking for rare. You know, even in RNA-seq or ChIP-seq or things like this, where it's sort of a quantification thing, which will degrade a little more gracefully. But you can imagine the dynamic range of those things goes down pretty rapidly when you start cross-contaminating samples. So for example, suppose you have a locus gene or something, RNA-seq or something. All the samples are high at that locus, except for one sample where it's off, the gene's off, right? Your manipulation is killed, you know, it's killed it there. But one sample is a 0, the other samples are high. Now you start mixing the samples a little bit, not even much, right? 1%, less than 1, you know, whatever, low amount. The high guys don't really know much, right? Because if you're mixing high to high, the little 0 guys only 1 out of 30 samples or whatever. The highs don't matter. But what happens to pore 0? Pore 0 starts getting a little bit of the other guys, 1% of the high. 1% of high and a 0 is a big change in dynamic range, right? So now maybe the dynamic range between low guy and whatever is much less reduced. Maybe when you do your quantitative PCR or something like this or whatever, you're like, I don't know, the separation is way better than I've seen in RNA-seq or something. It's better. It wouldn't take much in some cases for sample mixing to actually start, affecting the. Okay. Yeah. Okay. So there's basically 2 approaches people take this. Nobody really does it in the full alignment view, or maybe they should. So there's 2 major approaches, which I'll call basically the blind faith hypothesis approach, top-down approach. You know what you did on the wet bench. The kit documentation, you make your sample sheet because you know what you did. You submit it. You let the Dmacc, you don't even care what parameters it uses, whatever, right? You basically do exact match. Done. Whatever comes out, that's it. Answer problems, maybe, I gotta do a template, whatever. It's basically blind faith that everything's working. Because that some problems are just matters of degree and so on, and the science is down the bus or whatever. I mean, you can do useful science that way. I'm not saying you can't. But what I'm saying is, if you don't even have a way to know that problems are happening or what magnitude they're happening, you can't really believe that there aren't any just because you're not seeing them or looking for them. And you can't really believe that there's nothing to do in Dmacc just because you're thinking like that. It's just a point of view. So I will contrast that to this de novo empirical discovery resolution of empirical versus expected approach that I've been doing, where you basically let the data tell you what's going on, so that you actually know what's kind of in the mix in terms of just, what does the data say that the mixture components are? You don't know their sample identity, but you can at least say what is the indices that are there. Then you look at the sample sheet and try to match those two up. And ideally, that's basically perfect. And a lot of times, the basic is perfect. The most common problem is that people forget the force spikes and pi. And the effect isn't really a big effect. No big deal. That's so. Is that a problem or not? Well, it's the difference, but it's not. So no big. But there's a lot more space of problems than that. I mean, it's people can have missing samples, extra samples are not that uncommon. And how do you know they're extra samples? Oh, when they're like adjacent indices from the box, that's a high confidence, probably. When it's other indices you've seen from the lab before, probably real. Things like that. And sometimes you find out that people, you can say that like, oh, you have all these lectures, and then you go back and check their lab notes and say, oh, you're actually right. Whatever. So I mean, it's like, it's stage when we'll talk about the zoology of the problem. Each problem, some of them are more severe, less severe, and so on. And the rates of recurrence are always whatever. But I'm just saying that, like, like I said, so by doing this, one thing I learned is that what are those problems that occur? What are the triggers for them, and so on. And so one easy takeaway at the end of the workshop would be just don't do those things. Right? So then you don't have to worry too much about the actual process of discovering and dealing with trying to recover the lane if that's possible when you do have. You can simply just avoid the problem in the first place. But then, depending on how much you get into this, you may want to, get to those levels. And parts of this empirical approach are useful on their own, not too hard to do. And so it's not like a binary yes or no thing that you can kind of do this way or not. This is just another view and another thing that helps you have a better visibility to what's actually going on in your chart, which is generally, I think, a good thing. So yeah, some of the stuff we're going to talk about is fairly complicated, involved, and so on. If that's not your thing or whatever, just don't worry too much about it, because there's pieces of this that should still be useful, and you don't really have to do this. Commit to this approach and get fully into it to kind of like get some benefit from it. We talked about this a little bit before when I talked about the amplitude. Sometimes you can actually do much without using it. Some protocols actually do what are called inline indices. That's when you actually arrange the tags to be embedded inside the main reads rather than the index reads. In fact, in the days before we had index reads, you only can sequence with the sequence, which is the main read. But there's still some protocols that do that. There's just other things you can do, like alignment-based signals or other sequence features. Like I said, the Afghan libraries, where essentially the location of the genome to come from is already identified. When you have both, if you're doing inline indices, maybe that's the only thing you have or whatever. But when you have index reads and some of these others, that actually gives you some of the ways to actually prove that some of these things really are occurring. And they're not theoretical. Okay. Yeah, so here's the numbers that I kind of was talking. I need to get this little thing at the bottom out of the way. That's we'll put you up here. Okay. All right. So yeah. So another thing is actually is that we find one of the more benign things that we find happen in differences between is that the sequencer has systematically corrupted one of their indices in such a way that a large fraction of the recovery for a particular library is lost. And so there are cases where this basically demultiplication went okay. Maybe one of the samples has low representation, twofold, threefold, lower than something. And he's not right. But sometimes the answer is actually that the sequencer just systematically corrupted his index in a particular way. And relative to the mismatch, the Dmacc mismatch tolerance or whatever the tolerance of the Dmacc, the corruption variant is not matched to that sample. And so actually what you can do is realize that's happened with the vertical approach, add that as a new target or adjust the Dmacc tolerance, capture that guy, and now that sample actually rises up to kind of the normal level of the other guy. So I mean, in that case, the effect is sort of a little more benign in the sense that all the user's done is lost representation of the guy. But I don't think you want me pointing to random samples and filling half for the representation if you didn't have to, right? It's just sort of if it's just sort of an easy technical thing to recover that guy, then why not? So like I said, sometimes 10, 20, 40%, even the majority of a guy can be sequencing as a different tamer than what the or that I mean, it's possible that, for example, that would have to synthesize problem, which isn't generally perfect. So, I mean, adapters themselves could be a little bit wrong, but generally, the adapters are synthesized by probably of course between custom adapters. That's not really a serious way that some of these errors happen. So just because the intended index sequences systematically with different units does not necessarily mean that the original adapters molecule have the one thing. I think the majority reason why that happens. Base column topologies of the type of we already talked about the consequences of mismatches and so on. And what I do want to mention that the correct way to measure the impact is not in terms of the absolute number of wells on the whole flow cell. Typically, you want to measure the impact of these problems in terms of the size of the Dmacc of the guy himself, because representation varies so much across that. So, we talk about, oh, it's 10,000 wells, it's 100,000 wells, out of a billion wells. Well, if you're the top library with 400 million reads, no big deal. But if you're one of the guys on the low end and you're a 900,000 read library, maybe that's a big percentage, So you really want to scale these things relative to the size of the libraries that are being impacted, because they're not equally spread across the course. So here's a little thaw example to kind of you can appreciate kind of what might go on, which the numbers here are rounded kind of you may think can't be possibly this crazy. I think this is not that far away from extreme, kind of the higher end of what we actually see. So suppose in the absence of any errors, you've got two samples, A and B. The sequencer gives you, and Dmacc gives you 500 million A's in pile A and 500,000 B's in pile B. It's true, but we're pretending, this is wells truly from A, wells truly from B. So it's 500,000 A, 500,000 B. That's a 1,000. That's a 1,000 rate. So the percent of the pile that is placed in the right place, 100% here, right? There's 0, 0 here. Right? There's no mistake. Now suppose we miss Dmacc 1% of these guys. That means 5 million of this guy moved here, but 5K of this guy moved here. Now what's the picture look like? 495 million A's are still in the A pile, but now 5,000 B's are in the A pile. No big deal. The percent of the A pile that's actually from A is 99.999%. There are now 5 million A's in the P pile and 495,000 B's still in the B pile. And now only 9% of the B pile is from B. So that's not good, right? Not if you're trying to make any scientific conclusions of B from the B reads, right? The B reads, right? The reads you have in the B pile. So, yeah, as I say, am I really exaggerating here? Well, 1,001, that certainly happens. It's not the most common ratio, but obviously these kind of numbers all degrade as those ratios change and percents change. So it's all a matter of degree again. I just want to make an example that's kind of extreme, but not too extreme to be unrealistic and just to kind of bring up the point. But yeah, now what if I tell you that you're like, well, okay, how are you going to get 1% mismatch or something there? Well, maybe the index tag for B is only 2 mismatches away from the index tag for A. Does that change your view of the likelihood of that being plausible? Like potential in the realm of possibility? What if I make it an indel away? What about just a single mismatch away? 500 million reads with a single mismatch separating these two guys. And with the Illumina platforms, suppose the index reads have baseball errors like the main reads, 1%. Let's say you randomly distributed in random places. You're going to have an appreciable fraction, I mean, like a measurable fraction relative to the size of this thing come over. So that's the, this is why it's not really completely out of the realm of possibility. And then there's things like index hopping and so on, which are beyond, this is sort of a base call error as a source, but there's other sources like index hopping and so on that can also provide swapping. So if index hopping is occurring and you have high representation ratios, and basically high representation ratios are always a place for trouble, right? Because high representation ratios, you're a low rep guy mixing with high rep guys. Because of the representation ratio, rare things in the high rep guy become high magnitude relative to you. So weird stuff that happens to him, when you get unlucky and that weird stuff heads in your direction, you're in trouble. That's basically the lesson. Higher representation ratio is always sort of like the seed for trouble. And so the easiest thing that you can do to avoid trouble, don't set up for high representation ratios. I mean, sometimes it happens unintentionally, but don't intentionally, start scaling experiments for like, oh, I only need a little bit of one. Either find other libraries of similar depth that you can mix them together for really low rep cases using Hi-C or something, whatever. Try to, as I say, best thing you can do, try to keep all of your samples comparable together. The more you spread them apart in representation or composition or whatever, the more chance things are going to go wrong. Okay, so we are, so basically we already, without even really seeing much more, we already can start thinking of things that we can do ahead of time before we do this experiment. We're going to maximize our chances that in a way, we're doing all thought experiments so far. I think we've shown our data and how to do things. We can already sort of appreciate that there's something to make chances for things going wrong lower. So keep your indices far apart, right? When you choose your indices to attach to guys in your mix, keep them far apart. That means that if you're mixing with multiple people in your lab, across labs, whatever, it's such a complicated issue, you got to coordinate ahead of time. Right? We have a lot of cases where essentially people are post hoc meaning after their own library stuff, trying to find guys to mix with, right? Whatever. And people usually end up pushing the boundary, people usually end up squishing the indices a little closer than because otherwise they can't find mixed partners at all. Right? So the other thing to do is just try to get that stuff coordinated. And I'll give you a little web tool you can check the mixes and so on, make sure you don't get things that the entire mix is going to run in the lane. You need to check to make sure those indices are apart. And like I say, keep the representation ratios under control. Don't intentionally hit these big representation. What's big? Because it's not like every library. It's not like we even have the ability to really dial everything that perfectly, not with reasonable effort. And not all libraries really want to do the same depth. But 10-fold probably okay, 25-fold okay. Once you get into 100-fold and beyond, don't do it. If you want to, for example, you've got a lower spike in something, just use an Hi-C or something intended for that purpose. So we also already talked about this. I think I talked about this before I started the slides, sort of the kind of goals of this workshop here. Just general appreciation of Dmacc as sort of an important part of sequencing experiments and sort of underappreciated for the aspects of how to defend against it happening. That's probably the most important takeaways. Maybe we get you to the point where you can appreciate the different types of problems, the damages they cause them. A little harder is how to detect them, and a little harder is how to try to recover when it's impacted by them. An appreciation that the blind faith process doesn't always perform well, not like always. The skill, you can actually generally improve on that situation. Even if you're restricted to just revising sample sheets and using the same basic tools like detailed FAST2 or base-based. And just an awareness that there's this other approach, and that maybe it's a good idea to actually look at the main part and let the data speak for itself sometimes. And I think it's going to be probably too hard to get you to everybody to kind of fully do this fancier fancier approach, but like I say, it's not a really binary thing. Kind of you can pick up pieces of it, even just learning the lessons from it without actually being able to execute it. That has utility too. So anyway, the overall goal is just help you conduct more reliable, well executed, and successful sequence-driven science. Yeah. So anyway, yeah. So most of these lessons that I've been here, as I said, I've learned over basically doing I did about 600 lanes, pretty much about 600 lanes without the blind approach, without doing the data-driven approach for about 600 lanes. The reason I had to stop doing it is because it was essentially all of my time. It kind of was, sequencing, even, the amount of sequencing the core does increases over time, sort of exponentially. And the difficulty of this analysis approach increases super linearly with the number of samples mixed within each lane, which has also been going up. And so the combination of those factors meant that sort of originally what took me 20% of time in a week became 50%, 70%, 90%, 100%, and then went to above 100% in the middle of last year, where essentially I did Dmacc from morning to night, and it was fun. So that was sort of the motivation for this workshop, which is to, all right, that's not a sustainable approach. I can't do that analysis. If you did any sequencing with the core on the NovaSeq from when we first got it to about middle, early last year, you would have gotten a Dmacc report from me, which either you read or didn't, or appreciated or didn't, or whatever. But basically, that was me doing this approach I described on your data. And so the motivation of this workshop is to both try to make use of the lessons I learned from doing that, as well as to, maybe some people are particularly focused on quality or something, at least provide a way to start educating people on do that themselves. One of the big problems with the blind faith approach is it doesn't give you a lot of indicators things have gone wrong and things go wrong. Your only indicators are how much Dmacc fail you had as a percent. So like how many, in terms of the output of the lane, what percent of it were you able to assign the sample? And the other indicator you have is when a sample has what you think is an oddly small amount of data. These are really too simple. They're not sensitive to all the problems that occur. They're confounded with other things. At best, they kind of act like the check engine light. Basically, it just says maybe you should go to the mechanic. So anyway, so that's one of the problems is that there's not really always an indicator that things are gone wrong. And even if there is a problem, you're like, well, it's not that severe because I still get some data out. But like I say, some of these problems of index hopping, samples mixing, whatever, you can effectively have your samples mixed, even at high percentages in the file. Just going by the number of reads is not going to tell you. So anyway, so let's talk about myths now, right? So we've already hit upon some of them, at least maybe not by name, but in spirit. So these are things I think they're fairly commonly held, but they're basically all demonstrably false. So one is that Illumina baseball errors are drawing upon people's, say, main experience. They're rare, 1% or less, relative to the other two. Illumina baseball errors are rare. They're generally only mismatches. They're random. And they concentrate only at the ends of one. All ideas about what the error profile of Illumina is. And for index reads, those are all wrong. Not always in every lane to high degree wrong, but every one of those things is wrong pretty spectacularly in at least some lanes, like 5% of something or 2%. Well, in fact, actually, for this one, that applies to all of these. The baseball error is actually the rare mismatches, random, and concentrate only at the ends of longer reads. It's sort of really part of that. Actually, I'll take that back. For this first one, that's basically almost universally false in everything. It's just that it's usually not. It doesn't always kill you. So whether it moves on to the next level, causes to go down another filter, and that doesn't always progress to that point. So basically, that myth is sort of hard. for index reads. And for example, even if there are errors, say at least the majority of reads got it right. How could you do that? Well, that's definitely false too. You totally find cases where what is very clearly part of the sample sequences most commonly more, i.e. more than the expected sequence, some particular other sequence, like the mismatch. You can have a corruption guide beat the primary, no problem. That happens. Yeah, so this is really part of the myth of the blind approach, is that if you get an acceptable number of reads, with your a priori sample sheet and the default that Dmacc, all those samples must Dmacc fine and good sample sheet after everything. That's a more slippery one, but basically no, that's also false. Not as hard as false as the first one, but that's also false. We've already talked about this basically a little bit, which is the only real damage done by adding things together is reduced representation. Adding things can completely screw up the other thing. And we talked about this one. The commercial sample practices are well designed and optimized, and conflicts are rare. Actually, this conflicts are rare. This is also kind of a hard sell. You start missing kits even from the same manufacturer. When you haven't checked the conflicts, you may be surprised how many two mismatch, one mismatch conflicts there are. Okay. All right. So I don't know if you have any others yeah. So we've already actually talked about the kind of a little bit more in depth than just on this slide here, like what these boxes are, to talk about each one a little bit more in depth. But I think we've kind of already done that. Yeah, like this adding more things comes down to like tipping points in the base color is one way that can happen from effective index cycle complexity alteration. Yeah, so basically how a given sample, we lost it here, probably stepped on it. No, plug it back in. So the Zoom, we've lost the projector. Yep. It's actually the wall set. Right. So how a particular sample Dmaccs depends essentially on, among other things, how well the sequencing line went and so on. But in particular, it depends upon all the other samples in the lane and what their representations are, concentrations are as well. Yeah. We'll cover this when we talk about custom index designs at the end of tomorrow. Okay, so let's get into actual introduce what round folders look like, how to do stuff, and the beginning of how to get into the structure, how to start to this is going to be the ground colors and all kinds of things. So we're going to do a real example of the real one, Dmacc. It's been donated. You should do this one. Permission under two conditions, which is to delete all the moves to pick it up. So this is a 51-51 paradigm with eight multiplied single index. But I killed the maintenance the 51-51 down to 1+1. For our point of view, we don't try to do that. It's just because you can't really having the rest of alpha. Just keeping 1+1. So the lane was 51 to 51. Now it's 1. And there was a 20 tax expectation. The other condition was 3%. other than that, I can give you this run. So if you get on Hoffman and you look at use scratch, see focus in the workshop there, you will find what you'll pin down. Find it yourself. So I'll do a few slides. So we're going to start from the raw run folder as it comes off. The instrument talks to a Linux storage server that I run that provides Windows Samba, Windows SMB, CFS file sharing, essentially the RTA analysis inside of the instrument produces what's called a raw run folder, which is particular metadata files, plain text, XML files, other block files, and stuff. And then the base files themselves are described in binary files, format, which has changed many times since 2006. Mostly you don't have to care about all that stuff because we're going to use some tools, some developer, or something that's going to inspect the raw run folder for you. If you do care, basically I do know all the details for all the binary formats, for all the win file folder formats since the beginning. They're not that hard to use. They keep getting a little harder because the compression now and multiple files are grouped together now and so on, but basically they're still interpretable. Every single byte, basically primary files in the raw run folder are understandable. And basically the ACGTN decisions and per-base quality scores have been committed by RTA in those raw run folders already. There's no base calling that happens in DCL to FASTQ2 simply does two things. Takes out those ACGTN and Q scores and rearranges them into FASTQ files. It's just a format conversion. There's no computation of the base file or Q score. And just the Dmacc process in detail FASTQ can just control which file those things show up in based on indexes. But there's no recomputation of any base column. Base one does not occur in detail FASTQ. Once as the run comes off the instrument, the base column already been computed and forever, and there's no revisiting it. So yeah, so this run 51.851 means cycles 1 to 51 are the first n, 52, 59 are the first index, and 60 to 110 are the second main n. If you don't know the physical stuff, we'll get to the raw run folder in a second, and then we can actually look on Hopping too. But there's these two metadata files, one info.xml and one parameters.xml at the top level of the raw run folder. They will tell you what the physical root format was if you didn't know. The reason I want to mention that is because if you actually do start running and realize that sometimes what you requested and what physically happened, not necessarily the same thing. We might take your 51.851 and another user gives us another lane that's a 51.851. We're going to run the lane physically as 51.851 because that covers all the bases you asked for too. That has 8 extra. You didn't ask for I2 8 bases, but you're going to get them anyway. Because physically, when you do a flow cell, all the lanes on flow cell got to run in the same format. So we may increase the amount we run. A core will generally increase the amount they run to cover all the users that are mixed together on a lane. Usually, I think for most cores, at least for us, at least for the BSCR core, the lane is the unit that we transact in. I think we thought of, I don't know if that's happened yet. We were trying to do half lanes, but generally the lane is the unit we like to transact in. Because that's essentially what gets loaded. It's literally what's loaded on the machine. And for all the reasons we've been talking about, it's not straightforward to mix random people's stuff together. Anyway, so for us, it's like some, especially cores that do more library prep internally. So we're more of a research core where people have very diverse applications, custom applications, and so on, custom, very experimental stuff, as well as a lot of machine stuff. We have a very diverse user base, and so we don't do library prep ourselves, which also, that's a lot of hands-on work that somebody has to do. And we're not really interested in the business of doing sample prep. So we generally accept only already prepared samples, where some cores kind of you deposit raw material. Right? You bring them tissue from mice or something. They'll do the whole prep RNA-seq or whatever for you, whatever. And for those cores where it's sort of the preps are more controlled or something like that, then you may be mixing many people's samples together within a lane with less trouble because essentially you are the one person that's really doing everything. It's not really a mixture in the sense that we do. Yeah, okay, so now you can get these raw run folders for any BSCRC run on request by asking for something called live sync.
which is basically just an rsync to a particular rsync module name on a particular machine. And that actually gives you a live view into the run folder as it comes off the instrument. You can actually start those syncs as the run proceeds. You can then kind of do your syncs multiple times and catch up. And once you do one sync after the end of the run is done to get the complete thing, you're set. But that's always an option for BSCRC core stuff. And like, for example, people that use Cell Ranger, 10x and stuff, that's often their preferred way of getting it, because Cell Ranger is just a thin wrapper around BCL to FASTQ2, but it likes to eat raw run folders. As it's, most people feed it raw run folders as its diet, and that's how you get it. But we make these available for every run. We only keep them for a month though. Right? Runs are so big now, and so on. Retention time is guaranteed only for a month. Okay. Yeah, so raw run folders can be big, more than a terabyte for an S4, a big S4. It's format changed many times. We already talked about this. That the top level directory name will look something like this. Okay, you see this mess here. That parses pretty easily. The 21 is the year 2021. The 04 is in April. That's the 7 months. So that's April 7, 2021. That's the big start on the instrument, start date. The A00817 is the serial number of the instrument. That's literally the serial number printed on the plate of the instrument. 239 means it's the 239th run that that instrument has ever done in its life. The B means it was run on the B side of a B side, two flow cells per machine for the OC. And the H7 LGM BRXY is the serial number, literally a barcode printed on the flow cell. The run folder, so that's the name of the top of the run folder. The rest of the files in this thing, there's a whole bunch of metadata, which most of you don't care about. Instrument configuration files, logs, thumbnail images, you mostly don't care about. Then there's the actual, for the raw run folder, the FASTQ conversion tool, these are the key files in yellow here. That's the slox file, which tells it the layout of the XY coordinates of all the nanowells in the file. Because all the tiles are the same, you only need to do that for essentially one tile. Then there's the filter files, which essentially are the binary flags 0 and 1 that just tell you which are the wells that are marked PF1 and decide are singly filled. Because eventually, these days, we only basically, data is only reported for the PF1s. So to know which well is which, essentially, which subset those are. So you don't have to keep mentioning it over and over again. The rest of the files can essentially just use a linear order of files and use the combination of these two to define all the locations of where the wells are without mentioning them every time. And then the main event files here are these CBCLs, the compressed BCL files, which have the actual ACGT and I forgot the N base calls and per base the Fred Q scores, Fred scale Q scores. And for cycles 26 plus on the NovaSeq right now, it only contains data for PF1s. Essentially, the PF1 versus your decision made after the end of cycle 25. So these base call files, there's one for every lane and surface. It combines all tiles on a single lane and surface. And then there's one of those for every cycle. As I said, the base calling now operates kind of incrementally. That gigantic intensity matrix we talked about doesn't really exist all at one point. You collect enough cycles to sort of get things started, and then after that point, every time a new cycle rolls in, the base call will made for that cycle. Basically, a few cycles back. It's basically a rolling window of the data that's kept around, and the base calls in the middle of the window are being worked on as you go along. And so that's why it's called real-time analysis. So by the time the run is done, the RTA is almost done. By the time the run is physically done on the instrument, the RTA is almost done. There's this interrupt directory, which contains metrics and calculated statistics, which you can look at with an Illumina tool called SAV. And there's also some, there's probably a Python extractor or a C++ library for extracting data. It's beyond our scope right now to kind of show you what the SAV statistics look like. Probably kind of use SAV to ask some quality questions about the lane. Mostly those statistics are kind of stuff like FastPC or FastP might give you, but they're closer to the image analysis and the firmware part of it. So they kind of give you a little better view of those kinds of problems. But generally, for BSCRC core, you can assume that Suha or Manaz or me or whatever has looked at those. And if there's any really serious problems, we would have probably let you know or scheduled the link or we don't have failed rains are fairly rare now, but there are cases. One of the reasons why you have server contract lanes fail, they could be replaced with the agents. But you've really paid for them with a service contract, but you have to get a lot of failures before you're really making it back. But anyway, we are covered for lanes that fail. We can get replacements. Yeah, and then one little minor issue here is that if you live sync a rent folder, because lane delivery for us is on the lane basis, and we like security between our users. So if you didn't pay for a lane, you can't access the data for that lane because it's not yours, and you don't know what how sensitive the data is in any given lane in terms of science. Then what means is that generally our live sync rent folders only have a single lane data, whereas a rent folder has 2 or 4 lanes. The default for BCL to FASTQ2 is to try to convert the entire flow cell. And that's going to fail out with an error because you don't have all the data from the flow cell. It's like a partial transfer. So there's a command line option you have to give BCL to FASTQ2 that says, oh no, only try to do lane 1, only try to do lane 2. That's very important because you're just not going to have success with BCL to FASTQ2 unless you do that. All right, so Coffin 2 running BCL to FASTQ2 I'll show you that in the terminal window in a second. The resource requirements of it are unfortunately pretty high. There's not a really good reason for that. It's just the way they implement it that way. And it's not even like the way they implement it extra fast or extra flexible or anything. It's just kind of stupid. So RAM, tens of gigabytes or more, especially for the dual situation, which we'll talk about tomorrow. Yeah, for full grid, for large unique duals and large, what I call full grid, to look at index hopping, you can't have insane RAM requirements that are basically satisfied, like terabytes of RAM. And there's just no reason for it. Then you have to do other things, but let's not talk about that. It's basically the only standard tool that's around that does the raw run folder to fast conversion. So most people don't like to use non-standard stuff at this level of the experiment. So pretty much what everybody uses, even if it's not ideal. So I'm gonna ask for 3 cores, which is too small. 4 gig of RAM per core, 12 gig RAM on Hoffman for 4 hours. And even that's really small to run BCL to FASTQ2 and so on. But even that will make you wait unless you probably are in a research group to purchase some nodes on Hoffman. When you purchase nodes on Hoffman, you can use this Hi-C queue and get kind of more rapid reservations of 5 resource requirements and so on. So I will, because I want to not wait a long time here, I will ask for a Hi-C session. In fact, I asked for one at the beginning of the talk because I didn't know how long it would take to get one. But hopefully we have one sitting behind this window. But yeah, this could be a serious issue if you're kind of stuck in sort of the shared or campus pool on Hoffman. If you can, you need a Linux machine. Something with reasonable cores and RAM. So ideally, a machine that you don't have to have resource constraints on. So the job queue system on Hoffman is just kind of the way resources work. Controlled in Hoffman, it makes it kind of not so easy to kind of get a good session for everybody on Hoffman to do this. But anyway, the running of BCL to FASTQ2 does not depend on Hoffman or whatever, right? Generally, you just run it in an interactive session with something. And so on, it looks just the same, except you can do it on the local institution or whatever. No real. I just use Hoffman here because it's basically a Linux system that everybody has access to. So here I'm gonna ask for my session with the resource parameters, and then I don't really want to get into Hoffman and Unix resource limits and hard and soft limits or whatever, but just run these 3 lines. These just raise for 3 particular resource requirements, the soft limits to the hard limit. BCL to FASTQ2 is already installed on Hoffman. Obviously, you kind of need a new version of it every time some serious thing happens to a raw run folder format. So new instruments, serious new chemistry or something. Anyway, but this 2.20 version has been around for quite a few years now, I think. And I think there's been changes in a while for it. So anyway, so this has been around for a while. It's still the right one to use for the Dmacc 6000. And so it's in the module system already in Hoffman. We just do this module load. And then the command will be available. Okay, so now I need to kind of make this window smaller and get the terminal window open. Okay. Uh-oh. I don't know if we lost internet or whatever, but okay, we don't have a session. We did for some point, but then we lost it. Okay, so hopefully you cross the fingers here and we get one pretty quick. You can see my high P here. This is the request to use Pellegrini. I'm in the Pellegrini group, so there we go. Didn't have to wait long for that. Okay, so then we're going to run these little lines here. Don't really want to get into explaining. This asks the module system for what do you have with BCL to FASTQ in the name? You got that, so okay. And here we're going to run the tool and ask for its help. So there's the command line. There's the command line help for BCL to FASTQ too. And I think somewhere in here I must have somewhere in these slides, which I'll eventually distribute to you either tomorrow or the Wednesday or something. There's one somewhere in this slide deck. There's a link to the URL for BCL to FASTQ too. Okay, so here is the directory where my stuff is in the scratch space. And you can see here, this is the raw run folder, ZBP098. The ZBP099 will be our example for paired end tomorrow. So inside there, you'll see this is the top level run folder. And inside there, you'll see there's a bunch of files, like I was talking about metadata and stuff. Like here's the run info. Okay, we can look at that. And you'll see up here at the top, this is the physical format that we had here. So this is a 51.851. Or you could really look at the run parameters. That's also got the same kind of info in it. Here you can see that the run was loaded on the instrument as 51.851. The order they're written here is different, but it's still physical. It's SE I1 I2 PE. Planned was 51.851 and final was 51.851 because the run wasn't boarded, right? You can have runs that die in the middle so badly that the instrument just stops, not proceeding. What else? Okay. Oh, so most of the meat is in this data folder, and then the intensities folder. There's the slocks I was talking about. Then there's the base calls. Then it's separated by lane. Here are the filter files that define PF1, and then you'll see there's one directory per cycle. The C1 is first cycle of main end, and then I've deleted 2 to 51, and 60 is the first cycle of the second end, the second main end, and I deleted 61 to the end, because the user donating this lane doesn't want me giving you guys the main reads because it's just irrelevant, and I don't even know if this project is published yet or not. So the 52 here, the 52 to 59 are the index cycles. And so inside each one of these directories, you'll find, in this case, because this is an S prime lane, which is only imaged on one surface. I think the difference between S1 and S prime is that S primes are just S1s that are only imaged on one surface. I don't know, who knows? Maybe they're even made exactly the same and Illumina just puts a different label on them, who knows? This is the surface 2 BCL file here. If it was a 2 surface, if it was a top bottom guy, then you have 2 files here. So you can see that this collects all tiles for surface 2 in cycle 52 of lane 1. That's what that BCL file is for. Okay, so that's kind of what they look like. And here is the invocation of, first we're going to Dmacc twice at BCL FASTQ 2. So I kind of want to show you my bottom up approach as well. So we're going to first convert the run to just give us the index reads, basically, so that we can look at them and make some decisions before we Dmacc. Because we need, my approach is to not start with the sample sheet and look backwards with the starting data and we'll go up. So we need the index reads. I don't want to, I'd rather have the index reads and like FASTQ or something that's easy for you guys to play with rather than try to pull them out directly from the file. So first time we run BCL FASTQ 2 is essentially what I'll call the non-Dmacc. We're just going to put all the reads from the lane into one file. Not worry about demuxing yet, but in particular, we're going to get the index reads out, all the index reads. So that's the first goal. So this is the command line construction for BCL to FASTQ 2, and then there's all little. Let me explain the various parts here. This first parameter, create FASTQ for index reads, says that I do want the index reads as separate FASTQ file output. If you leave this off, it doesn't make index read outputs. For example, if you're doing Dmacc, the index reads are used. I mean, you've used them at that point. You don't really need them after that point. They've played their role. So you may not generate the index reads once you do Dmacc. In particular, also each read in a Dmacc fast queue in the main ends does have the index tag also embedded into the read name. So in a way, the base calls aren't even really gone, even if you don't have the index base calls aren't even gone if you don't. So there's just no kind of really point to them. In that case, and so when we do the Dmacc run, you don't have the at the moment. I do want the index reads because we're not Dmaccing, and I want to look at the index reads. So yes, please make me some index read fast queues. The sample sheet is nothing here. You know, just dev null, just empty file, whatever. No sample sheet. This is what tells it basically I don't want to do Dmacc. In fact, actually Dmacc is always on, but without a sample sheet, there's no samples in the. You know, there's just zero targets for Dmacc, and so everything with Dmacc fail basically. This is just logging. My little fancy shell redirects here actually just arrange it so that we can see the standard error and standard output as it runs and still log them to separate files. It's just some bash magic there. You shouldn't have any missing BCL files. I mean, here I've literally deleted BCL files, so there are missing BCL files. But when we tell it what cycles to convert, we will not tell it to convert the cycles I deleted. So there won't be any missing BCL files that you need to generate the output. So I shouldn't need this option. But anyway, if you're really stuck in a case where sort of like the files are corrupted or gone or whatever, you can throw in that option, and files that it doesn't have that it would need to generate an output base call, it will fill in with an N of quality score 2, which is the lowest it emits. This is just a gzip compression level. 6 ends up being a decent trade-off between compression time and space saved. Here I've only asked for 3 cores from Hoffman, so I got to stick to 3 threads. But this is way too small. So loading threads, 2, 3, 4, writing threads, 2, 3, 4, processing threads, 16, 24, 30, something like that. That'd be more normal. But here I got 111. I only asked for 3 from Hoffman to try to keep the resource requirements satisfiable fast. We're not Dmaccing here, so the barcode mismatches, which is its only parameter and only knob it has for Dmacc tolerance. We'll talk about more if it's when we do the Dmacc class what it can do, but the BCL fast can do is really limited into kind of what it can do and for the matchups, for the tolerance it can do for the reads to the sample sheet, canonical MER tags, and then these trimming lengths, because our sample sheet is empty. It won't try to do any adapter trimming, which I don't recommend doing in BCL fast anyway. But anyway, so all these parameters here basically shouldn't matter right now because no sample sheet, no adapter trimming, no Dmacc, so being solid doesn't matter. This is the key parameter that tells it I only want to convert lane 1. That underscore 1 means what? The lane 1. So this, if your run folder only has data for lane 1, you got to tell it only convert lane 1. So that's where you do it. And then this is the most complicated and key parameter. This is the mapping of physical cycles to cycles you want in your output, right? So like I said, you may have requested 51 and 51, but it may have gotten run as 51, 10, 10, 50, something. So the physical cycles are what they are. I'll show you in the metadata how to tell what that is. But then you got to match it up to what you want. So in our case, physical domain was 51, 8, 51, and I want 181. That's what I want right now. So you use a Y to say you want the thing as a main read. You use an I to say you want the thing as an index read. You use an N to say you don't want the thing. And you have to put commas between the physical ends. Basically, that's the rules. And then you should basically just make sure all the cycles are accounted for. There are other ways to use this parameter and so on. This parameter has extremely complicated syntax. But what I just told you, I think, is enough to basically do anything. And it's far more simpler to explain it that way. So what this means is 51 base is main end 1 physical, keep 1, throw away 50. Then comma, because we're switching out to index 1. 8 cycles physical, and I want them all. It's an index 3, I8. Comma, we're now moving to physical I2. Oh, there's into physical I2, sorry, in this one. So actually, we're now into the paradigm main read. Keep 1 base, throw away 50. So we will get 3 output files for each sample. In this case, all samples go into Dmacc file. So sample number 0. And so we will get 3 files. We'll get an R1 file, which is the 1 base main read. We will get an I1 file, which is our 8 base index, and we will get an R2 file, which is our 1 base paradigm file.
We haven't talked about UMIs yet, but if you wanted UMIs at this stage, you could basically just keep them as part of the index reads because there's no point. You're not demuxing yet, so you don't need to separate the idea between the UMI part and the index read part. They're all just doesn't matter. So you don't have to worry really about that yet. Okay, so while I'm talking, I should let this thing run or something, right? So here, let me actually, it's like we're gonna be like a cooking show. Right? I've got another copy already coming out of the oven, so you know, I won't have to wait for this to be done. But just to prove that, you know, I'm pasting in my thingy here, and there it goes. Okay, so it's running. Okay, so that's all the garbage as it starts up or whatever, and now basically you won't get any. It's working and it's making output. You're not going to really see anything until it's done. This is not a big fan of progress in the middle. It's more about blowballing you at the beginning and the end. So I'm going to make another Hoffman window here and just let that go in the background. I'm on the login mode now, which is going to be fine because I'm just going to be looking at a few things. I'm not going to get serious computation here. So, yeah. So this is what's being created here. This is the log files for standard error and standard out, and then this is the directory we specified. We haven't gotten to the end of the command line parameters, but anyway, these are the output directory and so on. It's making this directory. There's my 3 FASTQs in the Dmacc fail file being built there. Okay, so where were we? We were here. Yeah. Okay. Oh, then you got to tell it where the input is, obviously, and that you point at the top level run folder, the directory name of that top level run folder there, and then you got to say what directory you want the output in. So that's what I named it. And then it can also make more Illumina interop SASV files, more statistics, which really belong in the run folder alongside with the other interop stuff, especially if you're going to use SAV visualizer, they got to go there. But I don't like to contaminate the original run folder, especially because I'm going to run these multiple times and so on. So I'm not going to look at them anyway, so I just isolate them into a subdirectory of my output path. Okay. And then, yeah, so then here we go. So then these are our outputs going. Now they're not even done yet, but you know, gzip will decompress the head before the bottom's even done. So we can look at these, even though they're still being generated here. It's believe me, they're still being generated. It's just that there's buffering involved, so you don't see the file size growing by the second. But if we come back in a few minutes, these files should be bigger. But anyway, we can look at these even before they're being done generated. So okay, so you see the naming here. That's the sample name, which this undetermined means Dmacc fail. When we actually do Dmacc, your real sample names go here. This is sample number 0 on the sample sheet. 0 is the Dmacc fail file. Otherwise, they're just numbered S1 is the street on the sample sheet. I don't know why they do this. This is lane 1, index read 1, as specified by the use basis mask. Main read 1, as specified by the use basis mask. Main read 2, as specified by the use basis mask. This is 8 nerds for us, full physical 8. This is the first base of physical 51 for us of SE. This is the first base of physical 51 for PD for us. And then this is part number 1, which the way we run it, there's no file partitioning. So I don't even know if that's supported anymore. But in the old days, you could specify it like I don't want more than 5 million reads in the file or whatever. Maybe you're going to parallelize it. This is the part counter that just splits things into multiple parts. But these days, there's 1 part. So part 1 of well, we don't even know how many parts yet because it's not done yet, but for us, it's always going to be part 1 of 1. So that's what the normal ones. Okay. Oh, yeah. And so what's inside these things? We'll look at the index read because at least that's 8 bases. All right. So you've probably seen these before. I didn't ask, but everybody's run a sequencing experiment before, or what's people's background? Most of you nodding. Yes. How about the background? The back's quiet. Yeah, okay, everybody's. Yeah. I mean, otherwise, I don't even know what this looks like if you haven't done a sequence. But anyway, so these are the read main. FASTQ is a 4-line format with the 3rd line being useless. But anyway, this is the read main, and then that's the actual read sequence, ACGTNs, and then this is the Fred scale quality score shifted by 33 positions in ASCII, making quality score 0 the hash mark. Whatever. Anyway, these are capital S. They're pre-part. These are 30 or whatever. Figure it out, but it doesn't really matter for our purposes. Actually, that's the high end of the quality. the Illumina instrument for sure. So anyway, how do you parse these read names, right? So it turns out that this is the instrument serial number. It's on the nameplate of the instrument. That's the scan number. This is the 239th run that instrument is on its life. This is the flow cell serial number. This is the lane number. This is the tile. This is the X coordinate of the nanowell within the tile, and this is the Y coordinate. So the name literally tells you where this the well was. The next one tells you that this is the first end in this series. If it was, so it's basically, if the file name is I number, it's the number. If the file name is the R number, it's the number. So I1, it's a 1, I2, it's a 2, SE, it's a 1, PE, it's a 2. The N tells you that it's a PF1, but because NovaSeq only ever emits data for PF1s, it's always N now. In the old days, it used to be a Y, sometimes that tells you what the PF0 is. In fact, in the old days, sometimes you could still get usable data out of the PF0s, especially there. I mean, there were times when some percent of PF1 was 20%, but you could get a lot of usable data out of PF0s. Like I'm going back to GA1. Anyway, for a long time, so anyway, that's the history of that thing, but now it's always N. And then the next one is the control bit field. Back in the day when they thought they were going to embed like inline controls into libraries or whatever, and then have a base caller aligned to the files of that, and then they would mark up the read with what inline controls it had or something. A planned feature that died, and so it's a bit field that is always there. And then the last one is related to demultiplexing, but demultiplexing is off right now, so it's always a 0 in this case because we have no demux active. So that's that.
Now you understand FASTQ file is pretty completely. Oh, and I could mention the read order. The order is actually completely uniform, I think.
It's always, so obviously you FASTQ files is a single lane. So it's just how are the tiles and XY coordinates organized? Simply ascending by tile number as major order, then tie break by Y coordinate, then tie break by ascending. So that's why it's all 2101. This is an S prime flow cell. The old, like I said, the S prime flow cell is like an S1 flow cell that only images one side. The side that's imaged is side number 2. So the first tile is actually 2101 and not 1101 like everybody else. But anyway, this is basically the 2101 is the first tile on an SP. And then the lowest Y coordinate is 1000. So that's we're at the top left corner of the thing. You know, and we're working our way. It's basically just a raster scan on the whole thing.
So they actually are ordered basically physically in the instrument. But like I say, not all nanowells are PF1s. They're not all here. So, but anyway, but if you see if I scroll down and just hold down spacebar,
you know, eventually we'll work our way up into 30,000, and then we're on row 2, 1016. You know, 30,000 and 1031, 30,000.
you know, whatever. Anyway, it's just basically a raster scan of the whole thing emitting the PF0. Yeah, we already talked about that. Okay, now, one of the key things I think everybody should look at in every run, and one of the key things to look at when you're doing the empirical bottom-up approach is a histogram of the index cycles. So what I want is to know,
I want a histogram of these things, right? Using the index reads.
I want a histogram of those sorted by frequency, descending by frequency. Now you could load it, try to load an R or something. I don't recommend that, especially in an S4 run or something. These files are gigantic, right? Much more efficient to do this histogram in command line outside of R. So this is a little pipeline, which is a pipeline. I don't know how many thousands of times I've typed out this pipeline code. Anyway, but this is a very nice little example of Unix pipelines that does a histogram all in one little pipeline. So anyway, the gzip CD decompresses the gzip file on the fly. We're going to leave it on disk in compressed format, and this just gives us a copy out of standard output. I really recommend whenever possible you use pipelines with sequencing data. So keep all your data compressed as much as possible. Don't make temporary copies, uncompressed, whatever, scratch copies, whatever, intermediate files. No, just try to get your pipeline so everything just happens on the fly. So anyway, so this just spits out a copy of the uncompressed file without leaving the on-disk thing undisturbed. FASTQ is a 4-line format where X successive 4 lines is really one read. That's stupid. The paste here with dash meaning read from standard input, and paste operates in a round robin fashion. So this is read a line from standard input, 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 and those are the columns I want you to make. So basically line 1 goes in the lines 1 to 4 of the input file become line 1, columns 1, 2, 3, 4 in the output file. So we're basically, you know, we can just run this pipeline in pieces as we kind of build it up here. But here I'm just going to run that part and put it into less. Oh, because of the direct. That's assuming I never CD'd into the output directory. So it's referencing the path as a subdirectory of the output directory. So let me just back up one level so that we're in the right place relative to the way that little snippet is expecting things. So there we go. That's FASTQ turned into a one-line format so that every line just has columns now. With 4 lines in column 2 is now the sequence. So now we can just say, okay, give me cut off column 2. There's the MERS as a pure thing. That's nice. So then we want to sort that. Then we're basically going to pipe that to sort. Well, you know, sort with some decent RAM because sort's default buffers are too small. Okay, and then whatever. Okay, then that's going to look the same as this. It's just going to be in sorted order with a whole lot of rows that are all copies of each other. So then there's a here. Let me make just a demo file here. Let me just say like A, B, B, C, 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 C. Okay, so here's just a little. I don't want to wait for the sort yet. So, but here's a little file. Unique -C takes things like that and tells you just how many successive rows there are. So that says there were 4 A's and then 2 B's and 7 C's and 1 D. So that's once we do the sort, all the copies are in one place, so we can just count them. You know, we can just go like that. We don't have to find them all over. They're all gathered. So this unique would essentially construct the histogram at that point. And then we want the histogram itself in sorted order. So this is now would be in MER order, you know, lexicographically or ASCII-betically or dictionary order or whatever. They're all the same length here. So there's no real differences between those different sorting definitions. But well, actually, it's kind of stupid because unique. I think if you look at like the actual output of unique, it's got like a spaces. You know, those are spaces. It put weird. You know, it's like it's trying to make like 5, 6 digit like right justified numbers there or whatever. And just give us a 2 column. Just give us a tab separated format. That would be much nicer. So the first thing to do is just use a little awk or something and give me a nice 2 column tab separated format. Right? So now we got all the spaces we've gone. We have MERS in column 1, tab separated histogram in column 2, and let's sort reversed by the second column. That's the frequency. You know, that's now frequency sorted, and might as well tie break by the dictionary around the MER or something like that. Anyway, that's the idea. That's the histogram constructor. So okay, we got to treat this like a cooking show now because I can't wait for this. I think it's half an hour for that fast food thing to finish. So I got to pull out the. I got to pull out a here. Oh yeah, that's bad. Yeah, I just started writing files on top of it. I wanted to copy the one I had already into the done pile, but I just tried to copy them on top of each other while the other one's still running. That was great. So let's do this with the renaming. Okay, there we go. I don't really care whether this run that's ongoing right now is going to survive that damage or not, but it doesn't matter. We've got a copy. That's what happens when that finishes. Anyway, so let me copy that out one level here. All right, so D was our little demo like that. We're going to do that here. So this is same thing as what's going to come out of our current run, but is already done. We just in this we want to read from non-Dmacc 2 as the input now, because that one's ready. Okay, so that basically we saw how that pipeline works. We just need to run it now. Okay, so we're basically now constructing the index histogram for everything in lane 1, everything, the entire lane's index 3 for histogram right now. This should not take that long. It's an SP, it's not that big. If this takes more than 30 seconds, I do have a pre-computed version of this one too. Let's talk. Okay, this is what's going to come out, but we don't really have to wait for it there. But we'll check when it is ready that it did work, just so I prove to you it's things are working. But let's talk about what comes out of that. So here's the top of history. Okay, and actually you can kind of sort of now get the start of the idea of how empirical stuff would kind of start. At least is that, well, those are all looking pretty high frequency. This is, what is that? That's, we're expecting 20 samples and it's an SP and that's 32 million, right? That's 32 million, 31 million, 20, or whatever. And then, well, okay, then it kind of, it's slowly falling, but then it's like divide by 2, 6 million, divide by 2, 3 million. And then these are 600,000 and going down. Anyway, there's some kind of representational break there. And then maybe a transition down here. But, if there's any justice in the universe, that's the sample. So let's start. Now let me show you some. When we talk about okay. Let me just check. Yeah. Okay. So it was intended as a 20-plex, and there are 20 reds. That's a good sign. There's 6 million of this one. Okay, that's the 21st. Poly dark, 3 million of that one. That one maybe, so okay, let's, what is 20, 21, 22? Maybe this really starts to something else. This is really corruption of these. We now need to start looking at these possibilities, but we kind of, but it's looking good, but details to check now. If we had paired ends, I recommend histogramming I1 by itself, I2 by itself, and then pairs I1, I2. We'll talk about that tomorrow. If you had UMIs, you'll want to take those out before you do the histogram because they are randomized things that are separating the sample into smaller pieces, and you don't want those to do this decision. So you would insert a cut or something to keep only the cycles that are the tag, the Dmacc stuff, not the get rid of the UMI. But anyway. The sample sheet that the user gave looked something like this. Of course, I changed the sample names here. But there were 2 projects, and there were 12 samples in the product. And this was the sequence they gave. Well, okay, now it's like a big bunch of bookkeeping and stuff to check. But that's right. That's the next slide. Okay, but the first thing to check is just % of PF1. How efficient was the lane at using the flow cell? Because we have that number now. We can just add up. Was the histogram done yet? Oh. Okay, I'm gonna start. Oh, I'm doing this. I'm doing this computation on the login node, which is not a good idea because this is actually not a trivial computation now. Anyway, all right, we'll see. We'll see. We're pushing it now because that's actually computed for several minutes and it's a gig of RAM and it's using a bunch of IO and stuff, and you're not supposed to do that kind of work on your login node. But maybe since we're sunk cost fallacy, it's already so far along, we'll just let it go. Okay, I got to get it from here. So that histogram is here. So let me copy that one level. So there, this is the one that's already done. Okay, so there it is, and just to show you a little bit further along. So I mean, you see, there's sort of like it rapidly drops off and reaches some plateau, right? So it's like these are if not the user samples, are sort of high frequency corruptions of things or something. Then we quickly get into a realm where there's lots of sequence at relatively low things. You may not even appreciate it that like there's this much sequences that are showing up at low frequency out there. It's not like the index reads are all just like the intended samples. I mean, no, there's how many lines in this histogram? 25,000, right? So that's how many distinct MERS we got out of that sequencing sprint, which is just 20 intended indices. Just almost all those variants are very low frequency, right? But the first thing we can do is just tell how many spots were PF1. Just tell me how many reads there were in all that we got. And so we can just use a little awk there and just add up basically that second column. It's an easy way to do it since we've already made the histogram. We don't have to go back and just count the lines in the original file. It's just that I need to use the pre-computed version. Okay, so this is the number of PF1 spots on this guy, 496 million. From the chart at the beginning of this here, I told you that the number of raw wells on an SP is 638 million, blah, blah, blah. And so, getting back to where we were. And so we can compute the PF1 rate by just 100 times this divided by raw wells on the thing, 78%, which is pretty typical. That's pretty much right in the middle of the normal range. This is the histogram I showed you earlier, and that puts us right here. This is a very typical lane for PF1. If your PF1 is low, you're not making as much use of the flow cells as you should. Library concentrations, effective library concentration, that's, there's a difference because not everything that quantitates maybe can actually form clusters. The nanodrops and whatever, they see DNA, they don't see a properly constructed adapter attached DNA, right? They don't see it actually working in terms of Illumina. Maybe your insert lengths aren't right. The instrument can only handle, if you do very long inserts or very short things, these are preferred in different ways on the instrument. Something wrong, there's something wrong if it's low. If it's really low, then you can activate base call pathologies just from that, and there's just not enough data for the base caller to kind of auto calibrate. Really, really low, and the instrument will just abort the run. It can't even focus. The microscope can't even find spots to focus and stuff or whatever. Okay, so here, let me show you a web tool that I actually put together. Most of my work is actually done in Mathematica because Mathematica's had a notebook interface for like 30 something years. And so I've been using it since the 90s or whatever. And it's just what, I've been doing it for so long, that's what I use. But most people these days use web tools, R, Python, stuff like this. So I tried to put together some stuff that's more easily accessible. And in particular, this little thing I'm going to show you here, I put together is a web tool and doesn't need any coding or anything to use. So this is a little guy where you can just throw in sequences and it will make little distance matrices for you. And so this can serve a bunch of purposes. This is a tool you can use to check mixes before you run sequencing experiments that say, are these high separation like we were talking about? It can also help us analyze like these histograms and stuff like this. So all you do is you stick in raw sequences. I can make this endlessly more complicated and fancy, have sample names attached to them or something, whatever. But I just said, let's keep it simple. You just give in raw sequences, which are the index rate, the index things you want to compare, whatever. It just compares k-mers together. And then it just does all versus all comparison and either does mismatches only, which is the first box, or with indels allowed, which is the second box. Here, each mismatch counts as a 1 unit, and here each mismatch or indel pair counts as 1 unit. Or if you like, each insert costs a half and each delete costs a half, because since the sequences are all the same length, to make them, if you do a delete, you're going to have to do an insert. If you do an insert, you're going to have to do a delete to get the lengths back to the right much. So you might as well just count those as half and half. So let's, well, I think the default set I've got in here is Illumina TrueSeq 1 to 12 as 6 mers. And you can see the minimum distance here is 3 from those, which is okay. That keeps them all 1 mismatch recoverable. As indels, of course, they're not great. They're just too short. But let's feed it, say the histogram. Where were we here? Where we're thinking about, okay, those guys maybe we're more firm about, and then there's this transition zone, and then, well, I could throw in a couple of these guys at the bottom too. So maybe the bottom 4, we're not so sure about. So here, let me paste that into a Scratch text editor window. And the main reason for that is because another great trick to learn, at least on the Mac, if you don't know it, is if you hold on Option, you can do rectangle selections. So let me just do an Option rectangle selection there, and I can hit copy, and I just copied out the first column into my clipboard. And I can paste that in here. And boom. Okay, so let's look at the distances here. So actually, you can already tell that some of these are low in mismatch to earlier. This is left to right and top to bottom is frequency related, right? Because we sorted the histogram by frequency. So clearly the guys at the bottom here have low mismatches to earlier guys. So that makes them more suspicious as being actually you know, corruptions of the others. In particular, number 5 and say number 24 are only one mismatch apart. So number 24 is probably a corruption of 5, and so on. Well, anyway, okay, so the purpose of this index checker thing was more towards helping you design things ahead of time and checking for conflicts ahead of time. I made another kind of slightly fancier version of this thing called the empirical, whatever it is. It's on the next slide. All right, and that's the empirical Dmacc helper here. And this takes 2 column files, more like the histogram itself, with the frequency data, and then helps you make the decisions here. So for the maximum benefit, you need to also tell it the number of wells, the number of PF1s on the lane, because you're only going to give it the top of the histogram, not the whole histogram. So somewhere we computed that number. Here's the number. This is the number of PF1s on the lane. Actually, I think I made that the default for this thing because I knew this was my example. So there we go. Okay, so we told it how many PF1s are really in the lane. We've given it the top of the empirical histogram, and now it gives us a little that same report back to us, but marked up a little bit. So it tells us that like this first guy is 6% of the PF1s, and the cumulative to this point so far, of course, there's only one, so 6%. And the next guy is also about 6%, but now cumulative you're at 13%, and so on. So these are just adding that up there. And then the rest are actually designed to help you kind of make tentative little Dmacc decisions. So suppose we consider the first guy as a Dmacc target. And I do that by just marking him with a little asterisk. And now you see that we've got a green row here. And now actually what it's doing is that all the guys that aren't marked are getting Dmacced as if they were getting Dmacced around. So, and what it is that, well, if you only had this as your only target, the closest thing that number 2 could be is he's 7 mismatches away to number 1. Well, that's because number 2 is really a different guy. I mean, it's a different sample. I mean, he's just very far away, very high frequency. Number 2 is also a target here. In fact, actually, it means we can kind of just, oops, there we go. These are all, well, I mean, I can overmark too, but let's just, okay, let's just go ahead. We said we were pretty sure of all but this bottom 4 or something, right? So let me suppose we just marked all but the bottom 4. 1, 2, 3, 4. Okay, that's everybody but the bottom 4. Okay, and then this is what Dmacc would like. In fact, actually, none of the Dmacc guys match other Dmacc guys. None of the Dmacc guys match an earlier Dmacc guy with low mismatches, right? We already knew that from the index checker thing, this one, but this tool will also let you know that, right? So these are all 7. The closest it gets is 3 mismatches, I guess, here. So like number 17 is 3 matches away from 9, 12, 14, and 15. Okay, and now let's look at the boundary where there's 4 I didn't mark, okay? Well, actually, the first one I didn't mark is 1 mismatch to number 19. So he probably is a corruption of number 19. In fact, this already tells you that the myth of Illumina reads being random, random errors or whatever, it can't be true because you wouldn't see this, right? So this is basically 6 million guys that are all corruptions of number 1, of number 19. Where's number 19? That's here, right? So here, let me just copy these to a scratch space and make them bigger so we can see them. That's number 19. And then here's number 21, which there's a 1 mismatch to that. And we think number 19, actually, if you check the user sample sheet, number 19 is the one they were expecting. But here, this is the difference, right? So there's an A versus C in the third base. So 6 million guys, and then this is 6 million, right? So, and then this is what? What's the other one? 14 million, rough numbers. So 6 million, which is, let's call it a little less than half. A little less than half of the expected sequence has a 1 variant corruption. It's just all, somehow the errors are all concentrating on this. Whatever is going on in the mind of the base caller ends up making the third base go A to C for this guy. Like a lot. Like, if errors were random, this would not be, you could not have enrichment like this where one guy elevates like this crazy high. Just whatever. This completely already blows away from this one example of the idea that errors are random in index reads. And in fact, if you were demuxing this lane with no tolerance and doing exact matches only, so that this is your only target, you would miss this guy, right? You wouldn't have him. You just lost 14 plus 6 is 20 million, right? You could easily get 20 million out of this library. But if you miss this guy, you're down to 14 million, right? So there goes 25% of the representation that you could have had. So it gives you sort of the flavor of the kind of things that you can see this way. I mean, this is very, very simple example. And this lane is very, very good. This is really one of the best lanes we've ever seen. I mean, it's like I didn't want to start you with one of the really rich examples or whatever that's got all kinds of things going on. But just, you already can start to see that even in the best lanes, this kind of stuff happens. Of course, if you're demuxing with one mismatch tolerance, you're fine. This will be captured by one mismatch away from this guy. But anyway, correct decision is not to include that guy as a target. He's really just a corruption of number 19. But what about the GGG dark guy? The closest thing to the green before that, at this point, all the green has been seen. So the closest thing to all of our targets from that is 4 mismatches away, number 16. Number 16 is here. That is the GE-ist one of the intendeds. And he's got 1, 2, 3, 4 Gs there in an 8 more, so he's half G. But he's still 4 mismatches away. Anyway, maybe that one clears up when I tell you that phi x I1 sequences as poly G dark. And the core at this point in time was spiking in 1% phi x to every lane as a matter of routine. And so that's actually not an unexpected extra. It's not on the user sample sheet, but that's phi x. That's phi x intended mix, 1% spike in control. And so that's actually really a target, actually. Now, it doesn't really interact much with the blind Dmacc because adding it as a target, I mean, it was always there, whether you knew it there or not. Adding it as a target in this case doesn't decrease the minimum distance between targets. This is 4 mismatches to all the other targets already, whereas target to targets already reaches 3 mismatches. So this doesn't decrease the minimum distance between targets. And then the way BCL to FASTQ2 works basically is that as long as that tolerance threshold isn't crossed, you can pretty much add and remove samples and get the same results. So it's a very slippery thing to say the exact conditions are whether that's true, but essentially, most of the effect of not having this on the Dmacc to BCL to Dmacc sample sheet was simply not to get it as an output file. No real damage to these piles from not having it in the sample sheet, but it was a mixed component. It was there, and there are cases where it's an unexpected close conflict with somebody else, and whatever. And we'll talk tomorrow more about phi x and some of the reasons why we stopped doing this routine spiking and some of the reasons why we did in the first place. But anyway, but that's really another target. Logically, in terms of this empirical Dmacc, it's another target. It's a mixed component, and that is it's really should have been expected and on the sample sheet, but it just wasn't on the sample sheet, but it really was there. And then the next two are one mismatch number 16 and one mismatch number 5. And so it is starting to look like we've reached the point now where we're into the corruptions of others. I could extend that list of a little longer. Here's the index histogram again. And what were we at there? We're at the 463,544 was the last one we kept. 463, so that's here. So if I wanted to extend that a few more, that would be like these. So I just made the histogram a little longer, and the report got a little longer. But what you can see now is that all of these guys that are coming up are low mismatch to earlier guys. So unless there's another sample in there that's sort of low representation, it's much more likely that we've now sort of seen all the components, and we're now just going into deeper and deeper into the corruptions of those. So that was 7615. So that's let's really crank this up, right? So there's a much longer one. We're getting some 2 mismatched guys. That's a 2 mismatch still 200,000 times. I mean, if you work out sort of the statistics of what your kind of main read error rates are and stuff like this, none of these things should happen. Right? They just shouldn't be able to concentrate like this. And so this is really just showing you index-based skulls being quite different than main read-based skulls. Not that it's really having that much of an impact on us here. As long as we use, say, like a 1 mismatch tolerance or something, which we're going to do for this lane, we'll analyze why that's both a good thing to do in this lane, and we already know it's a good thing to do in this lane because of the example we were just looking at, where a guy that goes from 14 million to 20 million plus just because of a 1 mismatch guy next to him. So yeah, we're going to want at least 1 mismatch, just Dmacc, 1 mismatch tolerance here. That is actually going to be the maximum limit because the closeness of the index of the targets here will limit BCL to FASTQ2's dumb algorithm to that's the highest you can do anyway. But basically the analysis will be 1 mismatch is required, 1 mismatch is possible, 1 mismatch is sufficient, and that is what we'll use as our choice. And It's also would have been BCL to FASTQ is default and base space is default anyway, so everything's happy here, no big deals. But there's this kind of a logic to checking, and not in some lanes, all those checks don't agree, and there are different things that could have happened between base space and the blind side Dmacc versus here. But anyway, like I said, I got to start with a simple example. So anyway, so this gives you some idea. And actually, though, if you get down here, here's our first one that's kind of far apart. Poly A, 40,000, 4 mismatches away to number 7 and 15, even as indels. So that's the first one. If you had to point to sort of like the next thing that wasn't covered by this list of green right now, it'd be a poly A, which sounds a little funny too. And for reasons that I don't particularly understand, empirically poly A does show up kind of, it's usually much lower problem than poly G and darks and other things, but the poly A is sort of another one that kind of shows up for no real reason. Don't really know. That is usually one that shows up. There may be some, I don't want to say much about it, but usually it's at that point, it's a fairly small percent. We're down here into the 40,000s compared to the main guys, which are 30 millions right now. So there's a point where you just, enough. So anyway, okay, so where were we? So yeah. Now, if you had kind of made a mistake and kind of over listed things as targets, like suppose I had put this one as an extra, you'll still see that like it's marked as green, but it's still one mismatch to an earlier guy, number 19. So at that point, you kind of think, oh, maybe I won't mark him after all. I'm going to take him back out. So anyways, so this list of green right now basically is the data tells us kind of this is your list of targets. All right. Now there's the question of what are the sample names for these things? Well, now we got to look at the sample sheet, right? So now we have to resolve differences, right? This is going to be an easy one to resolve. Sometimes this side and this side not matching up very well, and the worst lanes I've ever had were like 20 kinds of problems all occur at the same time. It can take a day. I spent 3 days on individual lanes trying to figure it out, and sometimes it involves interaction with the user and all kinds of things to try to figure out what happened. But hopefully, those I call the nightmare. I think the 200 I surveyed over the last year, I think that was like 10 of those maybe. That's like 5%. So anyway, so the point is we have to match these up against each other. So you can write scripts to do this and so on or whatever. I mean, you can automate more or less of it. For these small cases, you can really just kind of do them by hand. Like I'll show a little bit of how even this one could be just kind of really done pretty easily by hand. Okay, we need this list of we need that histogram. So we're giving my histogram back. So what do we need? So the last one we need is poly G. Okay, so that's that, and then it's a little too big. I want it all to fit on the screen. Okay, that fits on the screen. And there's one I got to get rid of, right? It's this one is the one that's the high mismatch, one mismatch corruption that's actually not a member of the list, right? So that's actually our target list there. That's the empirical target list. Maybe I want it to be half the screen. And then here is the sample sheet from the a priori sample sheet. Okay. And all you got to do is let's take the easy cases, exact matches. We'll start with those. So here, I'll take this sample. Command E on the Mac puts that in my find and replace buffer. And then I will select this whole line, cut it so it's now in my copy and paste buffer, find what's in my search and replace buffer, matches that one exactly, and now I will paste the sample sheet line. Right, that's the matchup. So this is not a great, whatever. I mean, this doesn't scale very well. I'm not saying like this is the universal solution to everything, but this is an easy one. And I mean, when you get kind of into the flow and your keyboard shortcuts, this is, you can just bang these out, right? So as I say, this is an easy one. All these exact matches, no big deal. Sometimes you get your fingers ahead of you. Kind of like, just like becoming clearer, right? It's always very satisfying when you have like 150 plex And as you get towards the end, all the guys are just filling up the holes perfectly. And that last guy fits in and the puzzle's complete. And they're the ones where like by the fifth, even the fifth sample out of 100, you're like, oh my gosh, not looking good. All right, there we go. Perfect concordance, basically. You got an extra sample. It's 5x. 5x dark, whatever you want to name it, whatever. We'll make up a sample name. These are project names, like this. And this 5x, of course, here is credited to the mix and not either of the sub-projects because it was something that happened at the core level. It's not part of either project. It's a part of the process of making up the lane. Something like this. This is basically our sample sheet. It's not formatted quite in the right way yet, but that's basically an empirically derived sample sheet. Very close to the original sample sheet, to be honest, and not really we're not really making any amazing achievements here with the empirical process in this lane, but that's the kind of I'm giving you the logic here and so on. All right. Okay. Okay, so like, for example, it could have been, I'm just trying to make a little more utility out of this lane without doing additional lanes. Like we had that high mismatch guy, right? This is the 6 million one mismatch corruption of number 19. That's this one, right? Suppose this had actually sequenced much higher, right? Something like this, right? Whatever. And then what would have happened is which sample is going to die? It's number 19, right? Which is number 19 was this one. So which sample is that? So we would have been, like I said, this can happen where actually like the most common form can actually be a corrupted form. So we might have had this, and it's because this would have dropped out. This wouldn't be here because this is lower in frequency than this earlier one. Now the true one, whatever true means, would have been one mismatch away from this higher version. We would have probably empirically said this is the target sequence. And so this sequence wouldn't be in the list. This sample would have been unpaired. We might have been at this stage, right? We got one sample missing and one extra. But then we would look around and we would say, oh, this is one mismatch away from this. And we would realize that that's what happened. And so we would then say, so this is a case where, okay, we would bind this, but this is a one mismatch away from the expected, right? So if you want to label your samples this way or not, it's up to you or whatever, but we would do this. We would also have the choice in the analysis of whether we would actually keep the sample sheet with the higher variant one or not, right? It could go either way and depends on what other things are going on. For example, let me not really elaborate on that. And I'm just, we're trying to watch our time too, so I'm kind of limited to how much I can go into like all of our permutations and stuff. But okay, let's move on because I want to run BCL to pass Q2 in Dmacc mode and finish this example up so that day 2 is clean and don't have stuff bleeding over into day 2. So then we compose what I'm going to call the bare bones sample sheet. I don't know if if you guys have done sequencing, you must have had to compose a sample sheet because when you submit a lane, you do have to give us a sample sheet because we don't know what's in your sample, and we got to, for the most efficient way of using the instrument, the great advantage of the blind faith mode of operation is that it's automatic, right? The sample sheets get made ahead of time. They're loaded on the instrument. They're merged by the core, loaded on the instrument as when you load up the reagents on the instrument. The instrument, as it runs, pushes data to base space, which also gets the sample sheet. And as soon as the run is done, base space activates and does the blind Dmacc, and it's ready to go. And so it's just automatic and ready to go. Obviously, the empirical method involves much more hand-on work and so on. So that's the main disadvantage of it. Plus, you need the expertise to do it and so on, whatever. And I haven't even gotten into all the problems you can recognize and how to deal with them and so on. But anyway, so you must have already been composing samples. you probably did that in one of three ways. One of which is to use IEM, the Illumina Experiment Manager. That's one way to do it, which is like a software tool, or maybe they've got a web version or something. But basically, it's a piece of software with fill-in boxes and check boxes and drop downs and stuff, and is aware of a whole bunch of sequencing kits and so on and so on. The other way is maybe like an Excel template or something somebody gave you at some point, and you fill in rows and columns in an Excel template. The other way maybe is you're just using a text editor and just composing one. So depending on how fancy your template is, and definitely if you're using IEM, you probably have seen sample sheets with a lot of metadata. It's like all these sections at the top. I mean, obviously the main meat of the sample sheet is something like this, with sample names and sequences. This is not negotiable, right? You need the sample name and the target sequence, whatever. That's the meat of the sample. The rest of the stuff is all this fluff or whatever. But like there's all this extra metadata. One of the sections of which is adapters and stuff like this. Anyway, that turns out to be all basically unnecessary. You don't need any of that stuff. And so I actually recommend just using BCL to PASQ2 with sample sheet that's essentially just the bare bones, just minimal stuff. In particular, the biggest difference that ever will cause, the only thing that really BCL to PASQ2 really pays attention to in that metadata that actually can trigger differences in behavior is adapter trimming. If you do get sufficient data in the sample sheet, BCL to PASQ2 will attempt to find adapters inside the main reads and trim them away. I really don't recommend doing it that way. I really recommend that adapter trimming be done with a purpose-dedicated tool after Dmacc as part of your read prep before you do the rest of your downstream pipeline. Downstream depends so much on what you're doing, but maybe a generic downstream read prep pipeline would do something like check for overlapped main ends, do adapter trimming, maybe quality trimming. If the right end has a particular quality drop off, you throw away the base. Anyway, there are tools to do this, cut adapt and fast-p and whatever. Anyway, people do that. That's the place where I recommend doing adapter. Adapter trimming really depends on the downstream. Like some things, some RNA-seq or something doesn't even really matter. The aligner is just not going to generic situation is in alignment to the reference genome, won't have the adapter following the part that matches. And you're only going to count the read as +1 credit for its location anyway. The particular bases that are, as long as it gets close to the right place in the genome, the rest of the bases don't even matter. So if the little end part doesn't get aligned, the adapter doesn't really harm it. Whereas other things like de novo assembly or something, maybe that would start to be more of a problem. You're doing small RNA or something, and then the adapters are there because the reads are so, the actual meat from the organism is small. But anyway, different downstream applications just have wildly different needs in terms of how important it is to deal with adapters, what their tolerance is to false positives and false negatives, and so on. It's just a much more complicated topic that can be really handled with some sort of simple process. Not to mention the fact that it's often hard to get the adapter sequences right. So especially when you're mixing many kinds of sample preps within one lane or multiple people or whatever, the chances of sort of getting even the adapter sequences right at the top of the sample sheet kind of go down lower and lower. Plus, as I say, there's not really enough parameters there to really count for all the different downstream applications. The right thing to do is basically just not deal with adapter trimming at this stage. Deal with it as part of each individual project. We're almost done for today. I guess I'll have to probably start the actual BCL capacity run beginning of tomorrow, day 2. Anyway, so I basically just don't recommend doing adapter trimming with BCL capacity 2. If you're getting core FASTQs from a core and you see a ragged right edge in your FASTQs, it's already happened. Because usually the false positive, when you have a couple 1, 2, 3 bases at the end of reads, once in a while they're just going to randomly match the adapter anyway. And so if your default assumption for the trimming is to always trim, if you're biased in the direction of trim rather than not trim, you'll cut away 1, 2, 3 bases at the end, just just routinely, tons of false positives there. But anyway, sometimes for some applications that's the right thing to do, like BSC maybe or something, and others it's not the right thing to do. Whatever. So if you do see a ragged right edge, it means adapter trimming has occurred. Anyway, what I recommend is very simple sample sheets that be filled fast to. No chance you'll activate adapter trimming if all those sections are already. And just do your adapter trimming in a different tool. So so the core data for a sample sheet looks like this. It just has to be formatted a little differently. It needs to be comma separated and not tab separated. The columns need to have a label row. And then this bracket, data bracket, has to be at the top that just indicates that this is the section that declares where the samples are, as opposed to the other metadata we were talking about. There's a thing called a sample ID. So the data per entry is lane, which the core will handle if it's not, whatever. If you have the wrong lane, we'd fix it. The sample ID and sample name, it's very confusing why they have both a sample ID and sample name. And my best recommendation to you is just always make them the same. Just don't try to use that distinction. So just here you see my names are repeated twice. Then there's a project, which just basically controls what directory samples with the same project get put to a directory named with that project. So it's just an organizational tool. So anyway, so that you can make a project name, and then for single-based indexing, just a single index target. For paired indexing, you will have a second target mark here. Now, sample names, a lot of personal preference there, but these are long, but I kind of recommend sample names, something like this. I recommend actually encoding the core first, and then the run, which you can't do ahead of time, I know, but if you're running BCL2 yourself, you can do it because you already know what the run name has been assigned. And then the index, because actually the rest of this, the sample identity, is really an interpretation of the index. Now you may think that, how could that ever be wrong? But no, we find cases a lot of times, not a lot of times, but you have cases where somebody intended an 8-plex, 2 samples are missing, two extra indices. Well, those 2 extra indices are probably the 2 that are missing, but which is which? Whatever. Anyway, the identity of a Dmacc file is really the sequence that was used to target that file. The sample naming interpretation of that target is a separate layer. And so, yeah, I realize the convenience of having the sample names attached to your files and something like that, but realize that that's sort of a lower order importance and an interpretation relative to the true identity of the file, which is sequence defined. And so that's why I recommend actually the most important part of the name is simply the target sequence that was used to pull it out. But the rest is really an interpretation, which is subject to change. I've also, not frequently, but I've been involved in RNA-seq projects with, say, 40, 50, 60 samples. And by the time you do your PCA plots, analyses, and so on, it's very, very, very clear that samples 13 and 27 are switched relative to what lab, whatever, lab notebook and people, you really try not to make that mistake, but it's very, very clear in the data. There's no way that that guy and that guy, there's no way that the treatment for that guy is the treatment, but it makes perfect sense if they're swapped. So they probably are swapped. And so then, to not drive yourself crazy in dealing with the consequences of all your files and so on, much better if your primary identifier for the files is based on the sequence, which never changes, whereas opposed to the sample naming, now you got to worry about files before the swap and after the swap. And it just drives you nuts at that point in terms of keeping it straight. So I recommend that the target sequence as the primary identifier, put that first. If you want to have this info here, fine, project and sample, but realize that it's suggestive, not absolute. The reason for all the structure is to sort of, by the time your reads make it to SRA and NCBI or something, this is sort of a nice universe. This works for all of planet Earth, right? This identifies this read it identifies this sample in a nice way for planet Earth, right? Sequencing core, run, sequence. I mean, it's sort of a guaranteed unique system and so on. So I'm out of time for today. I'll stay another bit if you have questions that you don't want to ask with everybody here. First thing to do will be to feed that sample sheet, which I have a copy here. So here's that sample sheet, basically ready to go. Okay, just there's the contents, and the invocation to BCL2 will look very, very similar to what we've already had. There's very little to change. Where are we? Go forward. We have to do the Dmacc tolerance choosing. Oh yeah, that's actually a little interesting bit of R that we can do. But anyway, here's the command. Here's the eventual command line for BCL2, and it's basically the same one we've already seen. But the sample sheet dev null is gone. Now you just basically get pointed towards your sample sheet. But that's most of the. And here I said use one mismatch tolerance, but that's mostly what you need to change. Okay, so that's where we'll stop for today. I guess I will stop the Zoom.